have a balance, otherwise it will, backfire on you. So that has to be there. And then mangroves are one of the best for the carbon sequestration, which is the like a gold mine, we can say that way. And just because there is only 1 or 2 minutes, I would not like take more time. I will give time to our co-chairman, Dr. Majumdar. But before that, I will request our host that please mute the speakers, mute the mic of the people who are not speaking at that time when the people are presenting. Otherwise, there will be echo and other things. And second thing, I could not find Dr. Manohar, who is our reporter. Is he around? Because he is the one who has to write down the things.
Good morning,
so,
sir.
Good morning.
Myself, Nitish Ranjan Prakash. I'm working as a scientist at ICR SSIRR Scanning Town, and I welcome you all in this symposium. And first of all, I welcome Dr. P.K. Das, sir. He is ex-head Department of Genetics and Plant Breeding, Vidhan Chandra Krishi Vishwavidyalaya, Mohanpur, Nadia, West Bengal. He has worked on improvement of black gram and jute, and he has a long history of working in academics. He has guided several masters and PhD students. He will be the chairman of this session, which is theme one on the system approach for coastal zone development, agriculture, horticulture, and plantation crops, and their tolerance to biotic and abiotic stress. And we are into the session 1, which is on agricultural crop improvement, including biotechnological approaches, genetic resource management, abiotic stress tolerance. Next, I want to introduce our co-chairman of the session, Dr. R.K. Singh, sir. He is currently head of crop diversification and genetics section at International Center for Biosaline Agriculture, Dubai, UAE. He leads the research team studying crops such as quinoa, barley, mustard, safflower, and tree crops such as date palm and helophytes like saliconia and sauda, among others. He has started his career as an air scientist at SSRI Karnal, and later on he moved to ED, and after that he moved to ICBA. He had led the teams which has developed and released several salt-tolerant rice varieties, including various basmati rice varieties in India. He has published more than 75 research papers in international peer-reviewed journals over more than 15 book chapters. Next, I welcome Dr. A.L. Majumdar, sir. He is INSA honorary scientist. He has served in various capacities at Vishwabharati University, Santiniketan, and has retired from Division of Plant Biology, Bose Institute, Kolkata, West Bengal, India. He has also been the visiting associate and visiting scientist in several universities in USA. First of all, I request a few of our members who has just switched on their mic, so please mute yourself so that the voice will not be echoed, please. Dr. A.L. Majumdar, sir, he is the fellow of National Academy of Sciences, India, and he is also fellow of INSA, IAS, and West Bengal Academy of Science and Technology. His research area were plant physiology and biochemistry, abiotic stress responses in plants, and plant biotechnology. And in this session, Dr. K.K. Manohara, senior scientist, genetics and plant breeding from ICR Central Coastal Agricultural Research Institute, Goa, will be serving as the rapporteur. Next, I want to hand over the session to our honorable chairman, Dr. P.K. Das, sir. Sir, please unmute yourself.
That's right.
Thank you, sir.
Okay. Thank you very much. Okay. Now, okay. Now, shall we start right now? It is time.
Yes, sir.
Okay. Now, I will invite Professor Glenn B. Gregorio, Director and CEO, Professor, University of Philippines, Deputy Division Head, IRI, Plant Breeding Genetics and Biotechnology Division. He has received several laurels and conferred rank of academician as member of NAST Philippines 2018. With these short introductions, I would request Dr. Professor Glenn Gregorio to present his paper. His paper title, Worries of Salinity Intrusion in the Mekong River Delta, Vietnam. What innovative solutions can we offer through breeding for salt-tolerant rice? So, Professor Gregorio, please.
Yeah, thank you, Dr. Das. I first request that I'll be a co-host so that I could share my slides. I'm about to share my slide, but I'm not yet a co-host. So kindly make me a co-host so that I could share my slide. So greetings to the participants and organizers of the International Symposium for Coastal Agriculture 2021. I remember participating and speaking in this symposium some time ago when I was invited with R.K. Singh at Goa. That's a very nice place. And now I'm happy that I'm here again, sharing another topic on salt tolerance and especially the experience in the Mekong River Delta in Vietnam. I'm still waiting for my slides that I'll be a co-host so that I could share my slides.
Prakash,
Title.
can you allow Dr. Glenn to be a co-host so that he can share quickly?
Now you try,
Yes,
sir.
mentioned.
Now you try, sir.
Okay, thank you. So as I mentioned, my title of my presentation is on worries of salinity intrusion in the Mekong Delta in Vietnam and what are the solutions offered through breeding. At the same time, I'll be talking about policy support, about policies on to make this happen. In my next slide, I just want to introduce you to my institution. After 29 years at IRI, I joined another seed company, and recently, I joined the University of the Philippines as a professor, and now I was on loan to lead the Southeast Asian Regional Center for Graduate Study and Research in Agriculture. This is an intergovernment treaty to create a regional center for Southeast Asia, and this is, we call it SIRCA. So SIRCA is offering graduate studies, institutional development, research, at the same time, innovative solutions for agriculture. And when I joined it last year, almost two years ago, we formulated the new vision, our new 5-year plan. We call it the SIRCA 11 5-year plan. We saw the need to rethink how we approach things, particularly in terms of how we view agriculture, how we view food, and how we view food production. So just like the International Symposium on Coastal Agriculture, you have a topic on transforming coastal zone for sustainable food and income security. Clearly, there is a need for accelerated transformation on agriculture sector so it can fully maximize for human well-being. So essentially at SIRCA, we are asking the question if an accelerated transformation through agricultural innovation is necessary, as we are doing in the coastal areas. We call it attain. We strongly believe that the systematic and integrated innovations here would require a new mindset where farming is viewed as a sustainable agribusiness. That is in contrast to the traditional view of farming is merely about production. So we advocate about ecosystem thinking, circular value chain, future products and services, low ecological footprint, agriculture 4.0, and the need for the next agriculture and rural development transformative leaders for Southeast Asia and the whole Asia and beyond. That's why this is our overarching theme, accelerated transformation through agricultural innovation. So this symposium and CIRCA has the same mindset of how we will tackle the problem through transformation. In my next slide, I just want to show you these are some of the CIRCA's water-related initiatives, interventions in the past 5 years. Most of the interventions we done for water is mostly for saline areas, like freshening salinity areas or saline areas, implication of lye irrigation scheme for agroecosystems and household adaptation in Binh Dai district, Bến Tre province of Vietnam. The use of ICT to assess climate and land use changes, impact on hydrological responses and sediment yield for future use land use planning and sustainable water management. This is a case study in the upper Ma River Basin in the northwest Vietnam. And the third is climate change impact on rice production and adaptation, a case study of salinity intrusion in coastal areas of Mekong Delta, Vietnam. And lastly, but not the last, we are still doing something, exploring livelihood, vulnerability, and adoption of saltwater intrusion and drought in 2 coastal communities in Mekong Delta, a case study in Linh Hoi, Tong Commune, Tran De District, Soc Tran Province of Vietnam. So these are just examples of what we are doing at CIRCA, and I'm happy that we are doing this, especially I'm as a CEO and director of the South of CIRCA, we are doing something on salinity, which is salinity and breeding, and which is very near my heart. In my next slide, I just want to share to you part of my presentation. I'll be sharing about this policy, CIRCA policy brief we released on salinity and drought. Salinity and drought significantly affect rice production adapting good agricultural practices is a key solution. Some insights of Mekong Delta, Mekong River Delta of Vietnam. So I just want to say here that recently there's a big problem of salinity intrusion in the Mekong Delta, which is the high rice production area in Vietnam. So you can see here, I will just say the highlight of this one in my next slide. This is the situations of the Mekong River Delta of rice farmers in Vietnam. They actually salinization has already enhanced or exacerbated by several climate change related hazards, has been significantly affecting rice production in the Mekong Delta, Mekong River Delta. This is the major rice production region in Vietnam. What can farmers do to effectively respond to salinization and ensure farm production is maximized? So how would we do it? Actually, most of the farmers in the Mekong River Delta are generally aware of the negative impact of salinization in the coastal areas to rice productivity and profitability. But what is the solution? So what they did is really the massive promotion of good agricultural practices, including salt-tolerant varieties in cultivating rices in the coastal saline environment among farmers up to the level that it has achieved significant and wider strategic adaptation. So farmers are implementing adaptation practices of improving the yield through application of fertilizer and switching from local rice varieties, which is a long duration, to high-yielding varieties with shorter growth duration. And recently, many new varieties have been released for commercial production and that have tolerance to salinity, including other related stresses like flash flooding, along with the adjustment of the cropping calendar to avoid the drought salinity intrusion cycles that led to the increased rice production during the winter and the autumn seasons. In the next slide, I just want to give you the 6 among the relevant policy intervention. The following 6 are the rice varietal improvement related interventions that are suggested. So I'll just give you the 6 and I'll just give you some examples of some of the 6. One is actually sustained promotion and support of research and development. This is very important. Must be geared towards the development of wider adoption of integrated rice farming technologies that ensure high productivity and resilience, as always mentioned by our chair. The research and development system must be fully integrated with the concern of other relevant sectors. So we have to include other sectors, particularly community organizations and private players in the rice industry sector in Vietnam. I have been doing salt-tolerant breeding, developing varieties, but now I realize this is just a small component, because what is important, organizing the people, engaging the government, engaging the local government, engaging the industry, engaging the academe, the researchers, and give it to the farmers with the extension, with full support, including financial support. The second is more research on how to effectively identify and design innovative ways to improve the efficiency of rice production must be complemented with actual setting up of credit and insurance system for rice farmers, both formal and informal. Bottom line, money to support them. Awareness building on adverse impact of unsustainable practices such as extensive groundwater pumping and agrochemical application could also add to the improved adoption of the practices. This is to be sustainable. As mentioned, we have to take care of the environment, at the same time take care of the production of the people. So this is the very intricate balance. In the next slide, you could see here, I'll be giving some example of this. Among the intervention is the fourth is subsequent investment to further improve varieties that have tolerance to flooding, drought, heat, and salinity, and breeding program for new varieties, which most of my colleagues are talking previously and after me, while support to traditional rice varieties to thrive must be maintained. So we have to develop modern varieties, at the same time preserve the traditional varieties, which some niche consumers are needing it, and some areas could not be planted with modern varieties. So we need the traditional varieties in some areas also. And because these are the sources also of our tolerance. In the next slide, I just want to give you an example. This is a classic example. Dr. R.K. Singh is very familiar with this. This is just an example of our experience in Bangladesh. Yield of salt-tolerant variety, that is a Biridan 47. This is a Iri variety that we developed with R.K. Singh. And as compared to the Biridan 28 in 3 locations in Bangladesh. So you could see here that if you compare Biridan 47, which is a salt-tolerant variety, compared with a popular variety with Biridan 28, under low salinity, which is less than 4 dcm per meter, you could see here that average yield of 7.1, still high yielding under very mild salinity, but the Biridan 28 has reduced its yield. You could see here a yield advantage even under low salinity of 1.3 tons per hectare. Under mild salinity, which is 4 to 6 EC at the seedling stage up to vegetative stage, you could see here the average under 3 locations for Biridan 47, it is 6.7 compared for 4.7. If you click it, That's 22% for yield advantage under low salinity. But under mild salinity, you have a 43% yield advantage. That's under mild salinity. But under high salinity and the lower part, you could see here at EC 7 to 12 compared to EC 13 to 16, you could see here that the Bridan 47 has 5.4 compared to 2.7, which is a yield advantage of 2.7. If you click it, then you have a 2.7 yield advantage under higher salinity. Under low, which is 100% yield advantage. That's amazing. But under higher salinity, level 13 to 16 at seedling stage, the other one has 4.1, but the Bridan 47 is already 0. So this is just a very classic example of the potential of breeding or adopting salt-tolerant variety in the coastal area. But you could not adopt this directly because this is adaptable only for a certain area in Bangladesh. And the quality is the one they like. It's different from other countries, especially in the Mekong Delta. So that's why I'm saying breeding research support should be there to continually develop particular varieties specific in their area. In the next slide, I just want to give you another classic picture of how the varieties to compare this man from the other side. On the left side, you could see this is Bridan 47, 28, which is already affected by salinity. And the other one is the salt-tolerant variety Bridan 47. And you can see also this old farmer on the right hand. You can see them. The rice at his front is already the traditional variety, which is already dying under salt condition. At the back, you can see a happy salt-tolerant variety. On the upper part, you can see a classic example. This is a salt-affected area in Bangladesh. Actually, it's nearby a salt farm. You have salt there. And you have also, the rice here is already still growing under salty condition. So this is very promising, but as I mentioned, you could not adapt this directly because the condition there is different from other conditions, especially in the Mekong Delta. In my next slide, you can see here that fifth intervention, which is related as an enhanced application of modern technology and science-based recommendation, along with capacity development that will be critical for improved rice seed quality, innovative cropping system, pest and disease management, infrastructure, enterprise development, and that will ensure efficiency at the lower risk of the farmers. So you can see here, we have to apply modern technology, not only the variety, but other associated management, which I'll be showing you later. The next slide, you can see an example of it is another classic example where RK Singh is very much familiar with this. This is a study in India, productivity of using improved salt-tolerant variety and proper management practices in salt-affected areas. I'll not go to the details of this, but I just want to show you that under farmers' varieties management, they have a yield, this is about 16 villages. Under farmers variety, farmers management, it has a yield of 1.77 tons per hectare. Under farmers variety but improved management, it has increased to 2 tons per hectare, which is 16% yield advantage. If you have improved variety and farmers management, it has increased to 3 tons and it's 69% yield advantage with an average yield advantage of 1.23 tons. But the good news here, if you improve the variety with the improved variety and improved management under farmers field, it has an increase, a yield of 3.87%, which is 118% yield advantage, which is 2.10%, which is very high for a farmer. So you could see here, if you have improved management, you have only 16%, the improved variety, 69%. But if you combine both, if you have 118%, it's not 1 + 1 = 2, it's 1 + 1 = 3. So there's a synergy. If you use tolerant varieties respond better to improve crop and nutrient management, thus increasing the stability of the production. So that's why with here in the next slide, you could see here that to affect double productivity in the target saline-prone areas, the expected contribution of breeding and crop management, I would say, is germplasm or the seeds is, if it's tolerant, it's 40%, and the management is 60%. We have to credit the management for this one. In the next slide, you could see here that if you have the management, it means pest management, post-harvest machineries, and crop and nutrient management. In the next slide, you could see here that if you do seeds alone and management alone, so there will be a gap of 30%. So there must be partnership between the management and the breeders to make this happen. So you could see the synergy, you could see the partnership, what is important, or else you have a 30% gap. Next slide. You could see here as at the micro level, farmers can adopt good agricultural practices, giving emphasis on the use of climate change ready varieties in growing rice to effectively respond to salinization and ensure farm production is maximized. Next slide. I have a few slides more. Just to give you an example of some recently released varieties in Vietnam. So they are really very active. I think even RK Singh has been very involved in many of these released varieties. So we are happy that we with RK Singh was able to contribute a little bit for Vietnam as they also develop locally. So this is just an example. The next slide, I just want you another 7-step to higher rice yield based on IRRI, as we with RK Singh have been working together. This as we give the varieties, we give them also the 7 steps for higher yield. So I'll not be going to the details of this one. The next slide, you could see here, this is my almost final note that I will say that in the saline-prone areas, they have the double curse. You have double problem. One is because it's not more than double, actually. It's more than double. Low productivity. The other one, seed production must be in the non-stress area, because if the farmer will use the same variety, the same seed where they harvested from the stress area, the plants tends to be susceptible. Even the variety is tolerant because they have already some stress when they were produced. Farmers need to buy fresh seeds and not from their harvest. It should be, there's some effect already. You need a very healthy seedling under the saline areas. And we need more research and more policy support, insurance and financial support in these saline-prone areas, especially the emerging saline-prone areas. Lastly, to summarize, to operationalize the above recommendations, right at the farm level, there is a need for a strategic and wider adoption of good agricultural practices in cultivating rice in saline coastal environment. With these integrated technologies available, solutions to better respond to salinization could actually be realized. But what is critical, what is crucial, is a wider adoption among farmers and their timely, coordinated implementation in the Mekong River Delta. And I will say that partnership among the academe, research institution, industry, and government must take place to make transformation a reality and sustainable. I thank you.
Well, thank you, Professor. We are running short of time. Just to summarize things, you have suggested wide range of suggestions, policy priorities, and research on innovative technologies and use of climate change ready varieties. So thank you once again for your very thoughtful deliberations and a very practical way of doing things. And it does show that there is a crop development as well as integrated management that can create a really practical solutions for saline resistant type of breeding. Thank you very much. I would now invite next speaker. May I remind you once again that our time is very constrained, and please try to strictly maintain the time. Otherwise, it will be very difficult to continue in a schedule. So please cooperate with this. Now, I will invite Dr. Tapan Kumar Mandal, Principal Scientist, National Institute for Plant Biotechnology, IRI India. He is a pioneer to develop microRNA-based DNA markers of rice, received prestigious Genome Savar Award of PPFRA, New Delhi, for conserving salinity tolerant of rice. So, Dr. Mondal, please.
Good morning, sir. Is it audible?
Yes, sir. Yes, sir. It is audible, sir. You can proceed.
Okay, and you can see my slide.
Sure.
Okay, on the outset, I would like to thank the organizing committee for giving me this opportunity. So I'll be talking on directly, I'm going to the slides because the time is the limiting factor. So I'll be talking on the Oryza quartata, and I put the name Dhani because my farmers locally, they call Dhani in the Sundarban area. So I have put it. So basically, I'll be presenting some of the work which has been, which you are doing in my lab and which has been done in my lab. And some of the work has already been published, and few are then in the verge of published, not yet complete. So I'll be giving a summary of the work on which I'm doing the work on the Oryza quartata. So just to introduce very briefly, all of you know that it is a two halophytes and probably only halophytes among the 24 species under the genus Oryza, and it grows in the seashore up to the EC level of 40. What is not very known is that people who handle these, they told that it is seldom attack in the pest and disease. Apart from the salinity, it is also tolerant to the saline submergence basically, and to show the acid sulfide soils. So when we started this work in 2017, there was a very little genomic resources available in the NCBI. So we decided to go for the genome sequencing. Nevertheless, there was some wonderful work done at that point of time by the Bose Institute, Professor Rodundari Mujumdar, and as well as also Swaminathan Research Institute on Chennai. But they did the work on the individual gene, and then they saw the mechanism of the salinity tolerance of those genes. So with this background, my talk will be today in a 4 different parts. First, I'll be talking about very quickly the genome sequencing, which you have done of Oryza quartata for the first time. Then some cytological and anatomical study, and then we have done some LCMS study to know that what are the metabolites which could be responsible for this, and of course the microRNA of this particular plant. And all the results finally have been put to a database, and that is known as Oryza quartata Genome Hub. I'll show you that. So coming to that, in the 2017, the belief was that the genome size was 1.2 GB and it was a tetraploid. Although the same paper, it was written that it may not be the real size because size was based on the KMR or in silico. So we did the flow cytometry study and we found that it was not true. The actual genome size is 665 MB, much lesser than the actually which was believed at that time point of view. So then we developed, we generated different type of sequencing data. We used the Illumina sequencing as well as the nanopore. We used different shotgun sequencing, and then as well as different size of the parent sequencing library. And then we made the analysis to develop the quantic assembly. And then this is all the bioinformatical tools. And we use the Oxford Nanopore, which is a 3rd generation sequencing technology, to enrich the assembly. And finally, we got the assembled genome. And I'm happy to tell you here that the first time in the country, we use the Oxford Nanopore using the MiniION MK1B to decode the nuclear genome of this plant. And then we also did a de novo assembly to know the mitochondrial genome of this plant, and as well as the reference-based assembly to decode the chloroplast genome. So finally, we got the assembled genome, chloroplast genome, mitochondrial genome of the Oryza quartata. The idea was to decode, to generate the large-scale genomic resource so that we can know that what are the genes that are present in this Oryza quartata. Some of the basic stat of the genome sequencing is that estimated genome size 665 MB, but we could generate 570 MB. So there is a gap of something around 15% of the genome. Ultimately, genome coverage was 85.71%. Nevertheless, the depth of our genome sequencing was 250x, which is considered to be very high. So one of the important finding, we find it out that Oryza quartata is having nearly 20% repeat region in our genome assembly. So which is very, very less among the, not very, very, it is a less, quite less among the 24 species. And then this is some of the evidence which shows us how good our genome sequencing. So we have the 92.3% genome completeness, and BASCO reading shows that we have the 97.0% genome completeness. So these two stats shows that our genome sequencing is, draft genome sequencing is good. This is the basic measure which is used for doing any genome sequencing. We also developed a large scale SSR marker because that was one of our target, because this will be giving, ultimately helping the breeder to introduce the gene if the conventional crossing methods is being done. And we also dig out that there are something 4,000 plus transcription factor, which represent the 14.62% of total gene, which is present in the Oryza quartata genome. And we have finally annotated something around 33,000 gene, which is present in the Oryza quartata, out of which we are looking for what is the unique gene that is present in this. So we find out finally, after putting some criteria, which will show you, so 20 important genes are there, but out of 20, 11 genes were highly expressed under the salinity stress of Oryza quartata, which we are presently characterizing. And this is the, as I told you, that Oryza quartata is having the 20% repeat region, which is less among the 24 species. The reason biological significance is not yet known. Only the possible thing is we could guess it that the genome of the Oryza quartata, it is being the most primitive, has been expanded. And then, so as it is evidence that in different species, the repeat percentage has been increased. Much before our work, there was a paper published which also showed that Oryza quartata genome is having 35.1%. It is based on some in silico prediction, which further corroborated our finding that the percentage of the repeat region is less. So we also dig out the all non-coding region of the Oryza quartata genome. And finally, we find out that in the we made a dendrogram based on the 1:1 ratio of single copy orthodox gene, and we find out we are interested to know that Oryza quartata is having quite similar with the Oryza brachyantha, that is FF genome. So those who knows the wild rice, they can make it out. Oryza brachyantha and Oryza quartata is having similar architect, but it doesn't mean that they are genetically close. But here in our result, this shows that it is in between the BB and FF genome. So we also did some synteny analysis. Basically, the idea was if there is any region which is unique to this, and we find out that in Oryza quartata, synteny analysis we did with Arabidopsis and rice, as well as two other halophyte, that salt trees and extreme trees. And we find out that 2.19% of the Oryza quartata genome might have possible role because they are unique, and they might have possible role for the halophytic adaptation of this species, and we are working on that. This is the chloroplast genome, which is 134 kb, and this is mitochondrial genome, which is also 491 kb in base pair size, and we have done the details analysis of this. Finally, the paper was published with this data, but at presently we are generating the Hi-C data to make the chromosome level assembly, because our sequence is the draft level assembly. It is at the scaffold level. So we are now working on this to make it the chromosome level assembly. Now coming to this downstream application, many people are asking to us that we have, as I told, that we have predicted some of the genes which is unique to this, and these are the criteria. We put that certain criteria to based on that, and 11 gene we have find it out. First thing we did, we have identified two reference gene, because any functional genomic study, we need the reference gene which was not reported in Oryza quartata, and we found that actin and elongation factor 4 is suitable for qPCR analysis. Then we have identified two different genes from the Oryza quartata, and we are presently producing the transgenic Arabidopsis line that is in T2 stage, and we'll be characterizing those genes on the terms of phenotyping under salinity stress. In the meantime, we have find out the tissue localization and find out that one of the gene is located in the nuclear region, and other one is the nuclear as well as the cytoplasmic region. So this is the second application of the Oryza quartata genome. We are trying to develop some Oryza quartata specific marker. Finally, we have find out the 7 such marker, and one of the marker is very interesting. You can see this RM15008. This is the 1, 2, 3, 4, 5, 6 are different accession of the Oryza quartata, and rest of the different species of Oryza. And so you can find out that this particular two marker, they can produce the clear distinct pattern of banding so that it can differentiate from other. So when you look for this, what is the reason we found out that this marker, there's a 51 base pair deletion, because of which they produce the lower band, and which is not there in other species. So it means that this is very unique to Oryza quartata, and it has a practical implication that if we want to transfer the segment of the DNA from the Oryza quartata to other plant, this marker will be useful to trace back the Oryza quartata genome. And this paper was published in the 16th February, very recently. This was a collaborative work with the Sierra Rai Kotak. And then, so this was the first topic I have completed. The second topic is the cytological as well as anatomical study. So in the cytological study, this is very interesting, because Oryza quartata for a long time, it has been known as a tetraploid, and it is a allotetraploid, KKLL. So, you know, in the literature say that there is a many attempt to make a cross between Oryza quartata and the rice, and with a little success, although there are some sporadic report from Bangladesh as well as Professor K.K. Jana from the ex-IRI scientist. They tried hard and they could achieve this. So we thought that we will also doing some crossing. So we made some failure attempt for past 2 to 3 years. Then I asked the student to know what is the reason for this failure. That way the work was started. And then finally we landed up that to find out that the Oryza quartata is basically triploid. It is having the basic chromosome number 12, and 2n is equal to 3x is equal to 36. And these two are the independent. Here is the representative slide, but we have taken the accession from different region of Sundarban and Bhitarkanika of Odisha to confirm this. And we find out that it is a triploid. And we also, to confirm that it is a triploid, we took the pollen mother cell, and we find out that the pollen mother cell is having, as expected, many univalent, divalent, and other chromosomal aberration, as you can see on the pollen. So this confirms that this is a triploid, and which could be one of the reasons that the breeding success is very, very low. So we further wanted to know what is the status. So this was also confirmed through the flow cytometry, and we have made a karyogram of these 36 chromosome. We also wanted to know what is the status of the pollen. And very interestingly, we find out that, see, you can see this is the Oryza quartata pollen, which are mostly sterile. And this is the rice IR29 pollen, which are mostly viable. This is the 2% acetocarbonic test. And we also find out that the viable pollen percentage in the Oryza quartata is much, much more less than the IR29. And you can see, if you look closely to the seed, you can see this is IR29. This is Oryza quartata seed. Mostly 73% seeds are chaffy. That is one of the typical characters that it could be because of that the triploid nature of the Oryza quartata. And the seed which forms, very little seed which we form, we dissected, we wanted to see, and we find out this is a kind of pseudo seed, and it's having the black mass of the Giha seed. So this is also further confirmed that Oryza quartata is a triploid. So then we wanted to, there's another story behind it. We are doing some physiological study for some different purpose, and we suspect that it could be some C4-like plant, because the biomass growth of the Oryza quartata is much more higher than the other species of the rice. Then we find that the cranch anatomy is present, and you can see the bundle seed is surrounding the vascular bundle, and also the bundle seed is larger in size, and the mesophyll cell is very less in number if compared to rice. So the cranch anatomy is present. Then we are interested to know that whether the cranch anatomy is functional or not. And before that, I would like to say the vein density is also higher of the Oryza quartata. We also look for the organelle structure present in the bundle sheet. We find that the grana present in the chloroplast is non-functional, rudimentary, but the grana in the chloroplast present in the mesophyll cell is fully developed, which is a typical character of C4. So we wanted to know that whether this Oryza quartata is a C4 plant cranch anatomy is functional or not. We did the IRMA study, the carbon discrimination delta C13, but it has found, unfortunately, it was found that though the cranch anatomy is present, but it does not follow the C4 photosynthesis mode of this thing. So the biological significance we do not know at present, but we assume that Oryza quartata is very initial stage of adopting the C4 kind of photosynthesis. This is the general slides. We say that environmental stress such as drought, salinity, submergence, low CO2 force the plant to adopt the cranch anatomy and increase the vein density, high bundle sheet to mesophyll cell ratio. Oryza quartata is having all these three. Now the next step will be probably, if it goes, will be the C2 mode and then C4 mode. So this would be the reason, and enormous literature say that the salinity adaptation is more in case of C4 lineage grass family. So in that point of time, that Oryza quartata could be in the initial stage of C4. So this paper, I'm very happy to tell you that yesterday it has been accepted. Oryza quartata is a diploid plant with the initial event of the C4 photosynthesis evolution. Now, so I have completed the two topics. Now the question, basic question was, what makes the Oryza quartata is so tolerant to the salinity? So we did an LCMA study of the root of the Oryza quartata and the salinity state among many compounds. One of the compound allantoin, we find out its concentration is very high in the Oryza quartata root. We also did some kind of gene expression study, and we found that the pathway gene of the allantoin synthesis, there are two main enzymes here. That is, one is uricase and another is allantoinase. Both of their expression is very high in the Oryza quartata root. Assuming that definitely play an important role, but that we did a massive study on the allantoin content of the Oryza quartata and find that the root as well as the flag leaf, its content is very, very high. So we also did the enzyme study, allantoinase and uricase, and we found that the enzyme activity is also very high to the HPLC, and the allantoin content is also very high. So now we have cloned these two genes and we made the rhabdomycin transgenic. Now we are waiting for the phenotyping effect on that, and then we'll go for their promoter analysis to know actually what is responsible for this high expression. Now, finally, that we also did some RNA-seq and small RNA-seq study. We generated the library. We also did the degradome library for this, and part of the work has also already been published. And we are shortlisted some of the microRNA, novel microRNA, which we are characterizing through the transgenic mean. So this, as I told you, that although we made a lot of attempt for making the cross, but we could not achieve that. So this is in summary. Finally, I'll just show the database which we have developed, on which everything is there. Say like this. This is the Oryza quartata genome hub we have hosted for the end user, and you can go to any of the features on this. So you'll find the details whatever we have developed. We have put it into this database. I'm just giving one of the example. So in this scaffold, what are the different type of genomic resource that are there? And then in the download option, we have put the chloroplast and mitochondrial nuclear genome. We'll be putting very shortly. After getting the IC data, we want to put it the chromosome-wise level. So this is the chloroplast genome we have embedded in the background of the NCBI, and it is giving the details about the chloroplast and mitochondrial genome. So this will show the location of the gene of the Oryza quartata chloroplast and mitochondrial genome. So different publication which has come here, and also there are different options are there, so that you can get the information one can use for this. So with this, I think I'll be concluding my talk. So finally, the Oryza quartata genome size was determined 665 MB against the report 1.2 GB. And for the first time, we have done the genome sequence for this. Chloroplast and mitochondrial genome has been decoded, and find several novel salt-tolerant gene, 11 of which has been shortlisted, which we are characterizing for this. It undergo the process of adapting the features of C4 plant. It is a new finding from our lab. It is a triploid plant, and because of which the pollens are sterile. And when you dust the pollen from Oryza quartata to any rice plant, that could be one of the reason for the very, very slow progress of the conventional crossing. It has non-functional plant genatomy. Several salinity responsive microRNA has been discovered, which is underway for characterization. Allantoin might play the important role because under salinity condition, the allantoin accumulation is much more higher in Oryza quartata compared to the rice and other plant. And the repeat content is very, very low, lowest among the Oryza species. And so we believe with many other evidence that it is the most primitive plant, as we know, amphibian in nature, yet its genome has been expanded to the other species of Oryza species. So the genome size and as well as the repeat percentage has been increased to the rice and other Oryza nivara, Oryza rupipukon, all this plant. Finally, I have written the last year a book we have edited along with the Robert Henry, where the details of these informations are available. And I would like to thank my two previous directors who has encouraged me to do the work, and present director, my collaborator R.K. Rakesh Pandey from the plant physiology, who did most of the plant physiological work, and Dr. Midul Chakraborty from CRI Kotak, and my farmer's parent from who has gifted me this plant, because this plant grows in the Sundarban area. So we bring the plants from there, and all my students who are working on this in my lab. Thank you very much.
Thank you, Dr. Mondal. It is a very exhaustive study, and in a way, I can say that it is a dissection of Quarictata, and you have found a lot of different things, right from crunch anatomy. The question comes then C4, and then of course there is another problem you have started about a repetitive sequence, very less as compared to any other species. So all these things are very interesting. And what actually happening that you are trying to dig in very elite genes, and ultimately your objective is to use them in breeding.
Right.
And
Hello, sir.
Hello. I think Professor P.K. Das' voice has been cut.
I think it is something like a problem in his connection. So thank you very much, Dr. Tapan Mondal. And that's for the very exhaustive talk. And I think the time is constant. Again, we can just go ahead. I will request the organizer to allow another Dr. Gayatri Venkatraman for the next,
Please answer, sir.
and then
Tapan Mondal,
go
sir, please
ahead
answer.
with that.
Next, Gayatri Venkatraman, ma'am, please.
I've shared my slides with you. So do you want me to make the presentation using my slides, or should I wait for you all to make the presentation?
Yeah, please,
Ma'am, you
please
also
share.
can share, ma'am, no problem.
Yeah,
Okay.
let us
You also
do that, and please stick to the time, and that would be
Yes.
very important. And please, the organizer, please prompt when there is a less time, like a 2 minutes or something like that. Please prompt them.
Okay, sir.
Yeah. So good afternoon, everybody. Can I be heard?
Yes, very well.
Yes, ma'am,
Yeah,
you are-
thank you very much. So I think Dr. Tapan Mondal has given a very exhaustive presentation about why Cope data is important, or at least why it should be studied for its properties. So without going into too much of it, we are looking at a more micro depth in terms of what coarctata does. So what we've examined is sodium transporter function HKT15 in the halophytic rice Oryza coarctata. So now salinity, as already an introduction has been given, I'll just go past this. So essentially it is known that plants show a biphasic response to salinity. And what initially happens is when you apply stress, you have an osmotic response, and subsequently you have an ion toxicity response to the excess salinity. And this is achieved either by compartmentation of ions which are present by vacuolar compartmentation and or sequestration of sodium ions within specific cell types. So in this case, we'll be talking specifically of the HKT transporters. So HKT transporters essentially are of two types, and their classification is based on the gene organization and then ion selectivity. I'm just trying to make a rationale for why we launched this study and tried this out. So essentially type 1 HKTs are sodium specific uniporters, while type 2 HKTs are known to be sodium as well as potassium specific co-transporters. In A. thaliana, only a single HKT gene occurs, while in monocots and other dicot species, you have multi-gene families. And within the type 1 HKTs, the HKT15s are associated with salinity tolerance in cereal crops. So what is essentially known about HKT15 function in most cereal crops is that essentially they localize to the plasma membrane and they function in retrieving sodium from the xylem stream and reducing leaf and shoot sodium content. So as there's a radial inward transport of the salt into the root, it is at the level of the xylem parenchyma that once the salt enters the xylem stream, it is taken away from the xylem stream into the xylem parenchyma cells in the root, as a consequence of which the shoot sodium content and the leaf sodium content decreases. So most HKT15s show salinity specific expression. And in the case of rice, it has been shown that that while it is known that it shows remarkable adaptability in diverse ecological regions, and this has resulted in the use of the breeding material for salinity tolerance and the isolation of the saltol and the SKC loci, both of which happen to have the HKT15 transporter localized into it. I think previous presenters have already talked about how important the saltol locus is and how a yield difference is made simply by incorporating the saltol locus into local varieties. So I will not dwell on this. I will move on further into what we found out with coarctata. So the HKT organization, it is present on chromosome 1, and it essentially encodes what is called a 4 MPM domain type of transporter. It's a transmembrane transporter. Essentially, there are 4 MPM domains, and within which the SKC1 loci was identified, and the difference was attributed to 4 amino acid residue differences between Koshihikari and the salt tolerance rice Nona bogra. So essentially, while in the case of the saltol parent donor was Pokali, in the case of the SKC loci, this is switched, it should say Nona bogra. And in the case of the sensitive parent, in saltol, it was IR29, and for this, it was Koshihikari. So what is essential take-home message from this slide is that the allelic makeup which you find in Pokali and Nona bogra are identical, and the allelic makeup which is present in Nippon Bari and Koshihikari identical. And the reason I'm specifying is in subsequent slides, we'll be using the term Koshihikari interchangeably with Nippon Bari, and we'll be using the term Nona bogra maybe interchangeably with what is available in Pokali. So the data in terms of activity of the protein would be identical because the coding region of both of these genes in these are similar. So we've done heterologous assessment of transporter assays in Xenopus oocytes. And this is the data for the SKC loci by Ren et al. And what it essentially shows is that it is not only a sodium specific uniporter, it is also highly specific for sodium over potassium. But what is more interesting is when they've done the transporter assays, the Koshihikari variant is found to be less efficient in terms of inward transport of salt. So when we talk of inward transport of salt, it would be essentially sequestration of the salt into the xylem parenchyma, whereas the Nona-Bokra version, which is the NSKC version, which is shown here, would be more tolerant towards, which would be more efficient sequestration of sodium into the xylem parenchyma. So one of the set of assays which most people carry out is with the Xenopus oocytes. The other aspect which is carried out is expressing it in yeast in a sensitive strain and seeing what the response to overexpressing the ORF is. So as has already been mentioned, in the genus Oryza, 22 species are recognized, and Sativa and Coarctata fall on either end of the spectrum in relation with regard to salinity. So what we've done is we've compared HKT15 function in salt sensitive O. sativa and salt tolerant Coarctata. So already it has been mentioned that it is a highly saline tolerant species, and it occurs in the intertidal coastal regions in India and Bangladesh, and survives in a high range of salinity. So we found that OCHKT15, like OSHKT15, localizes to the plasma membrane. In addition, its expression is also induced under salinity in root tissues, but not so much in leaf tissues. So expressing OCHKT15 in a mutant strain, essentially this mutant strain is deficient in extruding sodium. So as a consequence of which, if you express HKT15, which should actually cause sodium to accumulate in the cells, what should manifest in the presence of salt in the medium is sodium sensitivity, and this is what we see when we provide increasing salt. So the B31 strain shows increasing sodium sensitivity when you increase the concentration of sodium in the medium. So essentially, it demonstrates that it's a sodium transporter. We've also examined OCHKT5 activity in comparison with the Nippon-Barre OSHKT15 activity. So the Nippon-Barre version would be the salt sensitive version, and it is not the Pokali version, which had 4 allelic differences. So the Nippon-Barre version here we are comparing would be equivalent to NSKC1. So what we find is it is has been demonstrated in the case of Nippon-Barre OSHKT15, OCHKT15 also has a salt sodium specific activity. But what surprisingly was different and news to us is that it shows low affinity sodium transport compared to even the Nippon-Barre version, which is supposed to be the sensitive version compared to the Pokali version. So not only does it have low affinity transport, it has 16-fold lower affinity for sodium compared to the Nippon-Barre version. However, at higher salinities, it is a more efficient conductor, shows a higher Na conductance vis-a-vis OSHKT15. So essentially what it says is at lower salinities, Nippon-Barre version is a better salt sequestration version. But when you increase the salinity in the bath medium, in the oocyte bath medium, the OCHKT shows better transport properties. So since we would expect that growing in a saline environment, HKT15 from a saline tolerant species should show better Na transport characteristics, we wanted to identify what were the modeling determinants which determined its low affinity transport. And this work was carried out in collaboration with Dr. Saudamani and NCBS. These Xenopus assays were carried out in collaboration with the lab in Montpellier, Dr. Anne Allen-Overy. And what we find is one of the critical determinants of this tolerance is a switch of a residue which is oriented towards the iron pore entrance. In the case of Nippon-Barre, it is a negatively charged residue which attracts the positively charged ion. But in the case of OCHKT15, there are other changes, but this was one of the primary determinants which was suggested by modeling to be one of the reasons for the lower affinity. So in the case of OCHKT15 near the iron pore entrance, we are looking at a positively charged residue which affects the which would affect the entry of the positively charged sodium ion. So what we did was we swapped the amino acid residues. Essentially in OCHKT15, we changed it to an E. So we would expect it to become a higher affinity transporter. And in the case of OS HKT15, we reversed it and made it a positively charged residue. And it shows the importance of the K239 in conferring low affinity sodium transport. So essentially, OCHKT15 has a 32-fold affinity in normal circumstances. Swapping the residue to a negatively charged residue increases its affinity 10-fold. Whereas in the case of Nippon Bare, which is around 2 millimolar, the affinity, swapping it into a positively charged residue reduces it by 3-fold. So essentially, why would you have O-Coke-Tata have a low affinity transport system living in an environment with excess salt? Essentially, the reason is in its ability to control the amount of salt which is present in its xylem sap. So the control here is Koshihikari, so which is why I mentioned that essentially it would have the same allelic makeup as Nippon Bare. So what we find is that at lower salinity, O-Coke-Tata accumulates higher salt in its xylem sap, which is in keeping with living in its in a saline environment where we expect it to be using the sodium as an osmoticum to counteract the effects of salinity. But what is remarkable is when you increase the salinity, you find that the xylem sap concentration is under much better control in the case of Coke-Tata, whereas O-Sativa is not able to keep such good control of the amount of salinity in its xylem sap. So essentially what it says is it's because that O-Coke-Tata has a lower xylem sap Na content, a lower affinity transporter would function better in spite of high salinity in the soil. So essentially, it seems to point to mechanisms that the transport of salt into the xylem sap seems to be highly controlled in the case of Coke-Tata, and therefore a lower affinity transporter would work. But where are the controls which control the radial transport of the salt into the xylem stream? Those aspects are areas of further research in our laboratory and in the collaboration with our partners. So this work has been funded by 2 DBT projects, and I'm very grateful to the Department of Biotechnology, as well as a short-term fellowship to one of our students who worked in the project. It has involved active collaborations between MSSRF and between BPMP in Montpellier, France, as well as NCBS, who did the modeling data, and University of Tasmania, who did the xylem sap analysis for us. Our collaborators are Dr. Anne Alenovery, Dr. Sadamni, and Professor Sharji Sabala. So thank you very much for your time. And if there are any questions, I will be happy to take them.
Well, thank you very much. Actually, because of a technical hitch, I was a little bit out of sync. But anyway, it is very interesting. It is another paper on Quark-Tata, the understanding of mechanism, how they control the sodium transport. And it is the affinity and the xylem stream, what is happening there. And this understanding hopefully will lead to us that how are you going to manipulate this for development of particular crop. So this is in fact a very interesting subject, and people are now understanding what is the mechanism and how it is operating in case of Quark-Tata. And actually she has given that lower salinity level and higher salinity level, what kind of balance is being produced there through xylem stream with the sodium transport affinity system. So this is very interesting subject. Dr. Singh, have you got any? Dr. Singh
So,
is
sir,
there?
You have already summarized, and then she is very well particularly talk about the HKT15, and that's I think this is the one of the most important thing on the salinity, particularly when we are talking about the seedling stage in the rice or other.
Yeah.
So I think because of the time constraint, let's move
Let's
to
proceed. That's right. That's right.
Thank you very much.
So,
and congratulations.
thank you very much.
Very nice.
Thank you. Thank you.
Thank you very much.
Dr.
Thank you
L.
very much
Majumdar
for
has
giving me an
asked a
opportunity.
question. Sir?
Anybody wants to ask?
Yeah, Dr.
But
L.
time.
has raised his hand. So please.
Yeah, Dr. Prakash, you can say that. In case if somebody wants to have the question, can you write in the Q&A with their email?
That's fine. That is better.
Q&A, question and answer, and with their email, so that we will respond and we will forward those questions to the respective speakers.
No, sir.
That's
Actually,
good.
Dr. A.L. Majumdar sir is also co-chairman. He was also like wanting to say something. I was
Sure,
telling you that.
sure, please.
Is it
Yes,
there?
sir, Dr. Majumdar.
Yeah. Okay. Am I audible?
Yes, sir. Yes, sir. You're audible, sir.
Okay. So what I would like to tell that this is one of the mechanisms through which the salinity tolerance has been worked out in Proteus quartata by virtue of one in particular protein and gene product, which has got a different kind of structure. And that structure allowed her to understand the mechanism through which the low sodium input into the xylem has been worked out. So this is, I consider, like the one with Tapan Mandal's work, that this is one of the mechanisms through which the sodium transport is controlled in quartata. So out of the mechanisms, this is one. And the next one, I expect that would be another mechanism for which the abiotic stress tolerance is worked out in quartata. So I congratulate both Tapan Mandal for the exhaustive work, as well as that of Gayatri, who has done an extensive work on the HKT1. And definitely many of the genes of quartata would come out to be having such products, which will explain why the mechanism is different rather than sensitive. That's all I wanted to tell. Thank you. And I congratulate Gayatri for the work.
Thank
That's
you, sir.
fine.
Thank you.
Thank you. Now I will proceed for Professor Manoj Maji, scientist, NIPGR, New Delhi, India. His research focus on molecular and biochemical mechanisms of seed vigor and viability during seed aging and storage. And he is going to give the role of different isoforms of protein repairing enzyme in repairing isoaspartate mediated protein damage in plants. And he is the recipient of prestigious National Bioscience Award, and he is a fellow of NACI and WAST. And his paper on protein L isoaspartyl methyltransferase, PIMT, a key enzyme for plant survival under abiotic stress. So, Dr. Manoj Maji, please.
Also, I would like to request all other panelists and other attendees, in case if you have any question, please write in the Q&A and write your email address so that we will respond you accordingly. Thank you.
Okay, so thank you very much, sir, for the introduction. And I would also like to thank the organizer for giving me this opportunity. And just let me. So can you see my slides?
Yes, very well.
Okay. So I'm going to talk about one of our work on this particular enzyme called protein A isoaspartyl methyltransferase and how it contributes to the plant and seed survival, particularly under stressful environments. So in our lab, we are trying to understand two very unique features of seed. One of them is how does seed stay alive for so prolonged period of time. I'm giving one exceptional example like date palm, it's known as khejur, which the seed can be viable as many as 2000 years. So it's an amazing quality. And even after 2000 years, date palm can able to germinate and can produce a successful plant. So how does seed stay alive for so long period? And there's another interesting features in plant that how does plant survive in adverse environmental conditions, even without moving away. So we're trying to understand these two mechanisms in our lab, and it's a complex mechanism. So there are a lot of mechanisms are involved behind this. So basically work on these three particular aspects to understand this mechanism of plant and seed survival, protein damage and repair, 26 potassium pathway, as well as protective metabolites. So today I will mainly concentrate on this enzyme PIMT, which is under protein damage and repair. And I will try to convince that this is one of the key enzymes for seed and plant survival under stressful environments. So I'll just briefly introduce what is this enzyme. So this is basically a protein repair enzyme. So it repairs isoaspartate mediated protein damage in cell. I'll try to explain in a very simple way how this works. So there are aspartate residues, and aspartate residues in a protein can undergo modification to form a isoaspartate. And this procedure and this formation of isoaspartate is accelerated by aging and stresses. So when the isoaspartate is formed in a protein, it affects the structure of this protein, so it affects the biological functions. However, in a cell, there is a protein repair enzyme called PIMT, which can recognize these isoaspartate residues and convert them back to the aspartate residue. This is the way PIMT can reestablish the protein structure as well as the functions. But this aspartate and asparagine modification is not random. It's basically there's some hotspot in a protein, like if the neighboring amino acids or next amino acid is glycine, serine, and histidine, then they're more susceptible to isoaspartate formation. And PMT catalyzes this conversion isoaspartate to aspartate. So this is how PMT works. So this PMT enzyme is widely distributed in nature, including E. coli, animal system, as well as plant. But this enzyme is mostly characterized in the animal systems as well as the bacterial system, but very limited studies have been done in plant system. And this enzyme has been shown to contribute longevity and survival of bacterial and animal systems. But in plant, when I started working in this enzyme, only few information is known. One of them is the PMT activity is highly abundant in seeds, and nothing was known essentially. So I started this enzyme when I joined at NIPGR and wanted to know the function and regulation of these plants. So we wanted to some of the key questions that we wanted to answer. Like first question, what is the correlation between PMT activity, isoaspartate repairing demands in organs and during seed development and germination? Does aging and storage accelerate isoaspartate formation, which may negatively impact the seed vigor and viability? And does environmental stresses accelerate isoaspartate formation accumulation in plants? Does PMT contribute in preserving seed vigor and viability by restricting isoaspartate mediated protein damage? Does PMT provide tolerance to abiotic stress? So these are the questions we wanted to answer over the years, and if so, how? So we basically want to know about the regulation and the role of this enzyme, particularly these two plants. So we have chosen these two plants, rice and Arabidopsis. Although we are also working on the Portia quadrata, but that's the beginning only. So first thing we have wanted to know the PMT activity in different organs in a plant. So here you can see that PMT activity is highly abundant in the seed. So why PMT activity is highly abundant in seed? Because seed contains very low moisture content, which is favorable for isoaspartate formation. So then we wanted to analyze the isoaspartate accumulation in seed, and here you can see in the seed the isoaspartate formation is also highly abundant. So the hypothesis is that the PMT activity is required in seed to limit the isoaspartate modification in seed. And so this activity is highly abundant in seed. So we wanted to know how this is regulated during seed development and germination. So here you can see the PMT activity gradually increases as seed development proceeds, while during germination the PMT activity goes down. So this is possibly because in dry seed the condition is very favorable for isoaspartate formation because of the deaeration stress as well as the oxidative stress. But when seed gets water, then the situation and environment become normal, so isoaspartate accumulation is going down. So as we know that during storage or aging, the seed germination efficiency and the viability is highly reduced. So whether there is any correlation between this reduction of seed germination during aging and isoaspartate accumulation, so we have analyzed the isoaspartate accumulation during the aging. So we have adopted accelerated aging or controlled deterioration test, which is regularly used for analysis of the aging test. So then we have analyzed the germination, PMT activity, and isoaspartate accumulation. Well, you can see that germination goes down as expected, but the isoaspartate formation goes up, drastically goes up. So it is basically detrimental for the seed viability. And PMT activity also goes down as during the aging. So this is very highly correlated that the reduction of germination efficiency upon aging or storage is strongly associated with the increased isoaspartate accumulation and reduced PMT activity. Next question, we wanted to know whether abiotic stresses also induces isoaspartate accumulation in proteins in plants. So we have checked the isoaspartate accumulation in stress condition, particularly oxidative stress and heat stress. And here you can see that during stress condition, isospotted accumulation is very high. And to counteract this high amount of isospotted, PMT activity is also induced during the aerobic stresses, which is basically required to restrict this isospotted accumulation. So in general, the PMT is encoded by 2 genes in a plant. So here you can see the PMT1 gene, PMT2 genes, and it encodes 2 full-length proteins, which has 1, 2, 3, 4, 5 domains in both the proteins. But in PMT2 protein, they have an extra internal extension. But besides these 2 isoforms, rice produces at least 6 PMT isoforms. Here you can see these are the 6 PMT isoforms, but some of them has lacking some of the domains. So we wanted to know whether all are biochemically active or not. So we've expressed them in E. coli and purified and then checked the PMT activity. Here you can see only 3 isoforms are actually enzymatically or biochemically active, which has all these 4 domains. So probably these are biochemically inactive, so they are significant. Still not why can produce these inactive PMT isoforms in plants. And we have also biochemically characterized in detail. So we have checked the Km value for the substrate and co-substrate, Vmax value, pH optimum. Here you can see they are fairly similar to each other. And we also wanted to know whether they are differentially expressed and regulated in organs or during seed development. Here you can see in the PMT2 is mostly expressed during in flower or seed, and during seed development they are fairly similar. But during the seed germination, the transcript level goes down in both the cases. We have also checked in their protein accumulation. Here you can see the PMT2, which is basically this one. So they are not expressed in vegetative organs, but they're expressed in flower and seed during seed development. And during imbibition also the like transcript protein amount also goes down. So what we want to say here that during seed development, the PMT activity goes very high, and dry seed retains these PMT proteins to repair isospotted damage during seed germination. And we also wanted to know in which part of the seed PMT protein is basically localized. So we have done some immunolocalization analysis, and here you can see in the embryo, particularly the pseudapical meristem, radical and polypterally, the PMT accumulation is very high, but it doesn't accumulate in the endosperm region, only the tellurian layer. You can see some of the PMT isoforms accumulation, but not in the rest of the endosperm. So it's basically localized in the embryo part of the seed. And we have also checked the subcellular localization. Here you can see that PMT1, as I'm saying, that there are 2 main isoforms in a plant, PMT1 and PMT2. PMT1 is mainly localized in the cytosol and membrane, while PMT2 is mainly localized in the nucleus. So there are 2 isoforms which is basically localized in 2 different subcellular compartments. So now, based on the expression analysis, you can guess that PMT activities probably is play a crucial role in seed vigor and longevity, because the enzyme amount is very high, as well as during stress condition, PMT activity goes up. So there is some possible role for stress adaptation or stress tolerance. So to establish this role or this hypothesis, we have generated PMT overexpression lines as well as RNA lines in both rice and Arabidopsis to understand or to establish the role of PMT. And here you can see in the transgenic rice, which is constitutively overexpressing USPMT1 or USPMT2, has a better seed viability. In control condition, that is in zero, in a normal condition, they have no differences in terms of germinations. But when you subject them to the aging, you can see only the transform line shows at least 40 to 50% germinations even after 10 days of aging of CDP. And we have checked this whether germination is also correlated with the viability. So we have checked the viability by tetrazoleum assay, and here you can see the transform line shows intense staining. That means they are more viable. When the staining is pale or light orange or light yellow, that means the viability is going down. So here you can see that the transform line shows improved viability as well as improved germinations. And next question, whether this improved seed vigor and viability is also correlated well with the isospotted restriction of the isospotted accumulation as PMT activities. Here you can see PMT activity is very high in transform lines, so their isospotted accumulation is less. So what we hypothesize here that high PMT means there is a less isospotted mutated protein damage, which basically helps to improve seed vigor and longevity. And we have checked also in the RNA lines where we have showed that less PMT in the RNA lines means there is a more isospotted mutated protein damage. Here you can see the isospotted is very high in case of RNA lines as compared to the control lines. and which also reflected by their core seed vigor and longevity. And we have also checked, since the PMT activity is also induced during seed during stress conditions, so we have checked whether their phenotypic analysis in stress conditions. So in control conditions, here you can see this is the wild type plant, and this is the transformed overexpressing Arabidopsis. This is the RNA lines. In control conditions, they are fairly similar, although there are bit improvement in the overexpression lines. But in oxidative and stress conditions, the control lines is highly affected. You can see by their growth. But in overexpression lines, here you can see they have improved growth, survivability, and yield. We have analyzed their number of calyx, number of rosette leaves, all are significantly better than the control, in the control lines or wild type and vector control lines. While in the RNA lines, here you can see their growth is drastically reduced. They are, even in oxidative and stress conditions, they even don't even produce to calyx. So they are highly compromised. So they are highly susceptible to environmental stresses when the PMT amount is very less. And we recently demonstrated that PMT also plays a key role in maintaining plant growth, yield, and survivability under salinity stress as well. So this enzyme can protect plants from different type of environmental stresses. So this is the correlation in a schematic representation. You can see where we can conclude that when the seeds has less amount of PMT, they produce excess isospotted accumulation, or they accumulate isospotted accumulation, which is adversely affected the seed vigor and longevity. That's why the seed exhibit poor seed vigor and longevity. When there's a higher amount of PIMT, then the isospotted accumulation is restricted, which improves seed vigor and longevity. Similarly, in stress tolerance as well, here you can see in the RNA line that has less amount of PIMT or less, which accumulate more amount of isospotted, because they don't restrict efficiently the isospotted accumulation, which eventually affected their growth and yield and survivability. While in the overexpression lines, when the PMT is high, they have efficiently repaired the isospotted formation and accumulation, which helps plants to maintain their growth and survivability under stress conditions. So what is the mechanisms? Why PMT mediated improvement occurs in plants and seeds? So there's a very common connection, as you can see, that if there's excess ROS, which is also known to restrict or known to affect seed vigor and longevity, as well as when there's excess ROS, it can adversely affect the plant growth and yield and survivability. So is there any connection between ROS and PMT mediated improvement? So we have checked the ROS accumulation in seed as well as in plant, and here you can see PMT overexpression line has less amount of H2O2, particularly in our stress conditions and under CDT, as compared to the wild type plant or vector control lines. So it is very well correlated with their improved seed vigor, longevity, and stress survivability. Similarly, in RNA lines, where there's a less compromised seed vigor, longevity, and stress survivability, they have higher amount of ROS or excess ROS, as well as the cellular damage. So we can conclude that this PMT mediated improvement is highly correlated with the ROS accumulation. But why ROS accumulation is less or altered in PMT or expression on RNA lines? Because this PMT is not an anti-oxidative enzyme. So we have checked because ROS is mostly neutralized or counteracted by the anti-oxidative enzyme. So does efficiency of this enzyme is altered in PMT or expression lines or RNA lines? So we have checked the activity of C8 cat and SOT in PMT overexpression and RNA lines, and we have observed that PMT overexpression line has higher cat and SOT activity as compared to the wild type plant, while in PMT RNA lines the activity is highly compromised. So why this enzyme activity is affected in PMT overexpression and RNA lines? So is there these anti-oxidative enzymes are susceptible to isospotted formation? So we have investigated this, and we have checked that why anti-oxidative enzymes activity are compromised in PMT RNA lines while increased in PMT overexpression lines. So to do that, we have bacterially expressed the CAT and SOD enzymes and purified them, and then do the thermal insult and aging, and then check the isospotted accumulation and do the MSMS analysis. Here you can see when we subjected this purified protein to the high temperature or aging during or thermal insult, or it's also considered in vitro aging, you can see the isospotted accumulation is more when it's treated by heat. And then we check by MSMS analysis, we have identified this aspartate 13, 27, and 29 of CAT2, aspartate 411 in CAT3, asparagine 102 in SOT, and asparagine 201 in SOT is converted to isospottate. So this isoaspartate basically adversely affect the enzyme activity. So that's our next investigation. So does isoaspartate formation affect enzyme activity of these enzymes? And can PMT repair isoaspartate formation and restore the catalytic activity of CAT and SOD? So we have also evaluated this, and we have done an in vitro PMT repair assay where we have used the thermal insulted CAT and SOD, and in one set we provide PMT along with co-substrate and other requirement for the enzyme catalysis, and in another set we have without PIMT, and we have used the purified thermal insulted protein and check the isoaspartate accumulation as well as their activity, catalysis activity. So here you can see without any treatment, if this activity is 1, and when we thermally insulted this protein, here you can see the activity goes down, catalytic 2 and catalytic 2. But when we provide PIMT in the system, you can see the catalytic activity is, re-established. So that means PIMT activity can re-establish the CAT and SOD activity, which goes down due to the isoaspartate formation. We have also checked the isoaspartate content of this protein, catalase and SOD protein. Here you can see when we provide CAT and SOD, thermal insulted CAT and SOD with PIMT, and then check the isoaspartate content, the isoaspartate content goes down. So what we have concluded here that PMT not only repair isoaspartate accumulation but also restore the catalytic activity of antioxidative enzymes. So what we have basically overall what we want to say is that PMT mediated protein repair system is critical in maintaining ROS homeostasis during aging or stress, apparently by protecting the catalytic competence of antioxidative enzymes that are otherwise affected by stress-induced isoaspartate formation and play a critical role in stress tolerance. So this is how PMT works. So PMT activity goes up in seed as well as upon stress, or upregulated during seed maturation or during stress condition, which is required. And so when the seed has a robust and efficient PMT repair system, they have a less protein damage. And that means when there's a less protein damage, there's a improved seed vigor, longevity, and survivability under stress conditions. So what PMT does here, so basically PMT protects, as we have shown that it protects antioxidative enzymes activity by repairing isoaspartate mediated damage, because this protein is very important for maintaining seed vigor and longevity as well as for stress tolerance. So those proteins can be modified to isoaspartate, which can affect their function. So PMT is required to protect them to yes, so PMT not only improves the seed vigor and seed storage life, but also improves plant antibiotic stress tolerance and yield by actively repairing deleterious damaged proteins. So we have only identified the antioxidative enzymes which can be susceptible to isoaspartate formation. So there may be a large number of proteins which can undergo modification during stress condition, and that's why PMT proteins is very important to protect them so that they can function during stress conditions. So the exploitation of PMT mediated improvement of seed vigor and longevity and stress tolerance in crop plants may have a huge impact on the agriculture economy. So on a single enzyme can protect seed vigor and viability as well as improve the seeds stress tolerance property of the plant. So these are the students. Nitin has done mainly what I have told here, and I also acknowledge the funding agencies, NIPJ, Port Grant, and DBT.
Thank you very much. Now this is another piece of paper which deals with the mechanism and where the particular enzyme system is responsible for both regulating the viability and longevity and its expression level, that is during different stages of development, is so important. And in doing so, it has been said that ROS mechanism, how that antioxidant enzymes, they are being affected and they are regulated by this PIMT. And there is a propositions that this PIMT on salinity also, there is a question of that.
Yes,
so
we are also working in Potassium quark data, so that work is being done. So probably
okay. This is a piece of your work here. So naturally, it is a very interesting piece of work. And again, we can clump together 3 different papers so far presented, and then it will give you some good idea how this salinity mechanism is operating. And probably in future, we will have much more clarity about these things. thank you very much. With this
Thank you, sir.
deliberations, I'll once again thank you, but we are running short of time. That's why we cannot entertain many questions. As I already suggested, that please send your questions and answers will be sent back to you. So thank you once again. Probably as per the schedule, there will be a 20 minutes break. Am I correct?
Yes, sir. Yes, sir. We will have 20 minutes break, and I hope we will assemble around 1:40.
1:40. 1:40.
Yes.
So the next speaker is a Viswanathan Chinusamy.
Yes.
That's right?
Yes, sir.
Okay. Okay. So 13:40 hours. That's
Yes.
right?
Yes, sir.
Okay then. So let us relax a little bit and then we'll proceed for the next. Thank you very much.
Thank you.
Any
Thank you very
comment by
much.
Dr. Sen?
No,
Okay.
it's okay. Thank you very much. I think Dr. Manohar, I can see him now. I think you are taking good notes on that because Dr. Das and we all will be dependent upon you as a reporter, you know. Thank
That's
you very
right.
much.
Okay. Okay.
Good afternoon, sir. Shall we start session? Sir.
ওই জলটা নিয়ে তখন থেকে খাতে
Please unmute yourself, sir.
এখানে রাখা পর্যন্ত স্কুলটা আছে
এইটাতেই নাকি অন্যটা জয়েন করতে হবে?
Hello. Is it okay?
Yeah, I think we can resume that.
Okay, okay. Thank you very much. So our next speaker, Professor Viswanathan.
I think he has not joined yet.
He has not yet joined?
Yes, sir. So
Is
We
there?
can swap. We can swap with next one.
Okay, okay, that's fine. So we'll go for oral presentations. There will be 5, 6 oral presentations, 7 oral presentations, and okay. So we'll first invite Dr. V. Dhanuskodi and others, and Dr. Dhanuskodi, presumably who will deliver the lecture, is from Krishi Vigyan Kendra, TNAU, Tamil Nadu, and his title, Evaluation of Different Salt-Tolerant Paddy Varieties in Sodic Soil and Farmers' Preference for Adaptation in Tiruchirappalli district in Tamil Nadu. So, Dr. Dhanushkodi, please. Is he there?
No, sir, I can't find him also here now.
Oh, not yet.
Not yet, sir. I hope they are busy in their lunch and then.
Anyway, now can I invite next one? Dr. K.K. Manohar.
I
Is he
hope
available?
not yet joined. I think that we have just only one option to wait, or else I am presenting.
I think Dr. Manohar was there.
Yes, sir. Yes, sir. He was there, sir. But maybe we have given very short amount of time. So
Okay.
What is the next option?
next option. Sri Devi A. Jakrel, madam is there. So maybe she can present her oral.
Who?
Sri
Veena.
Devi A. Jakrel.
Oh, okay, just a minute. Let me check it.
Madam Sri Devi, are you there?
I do not know. What happens?
She is mute and she has not yet responded. So that's why I was asking.
Veena Dineswaran, Vigneswaran. Nobody is there. Arti Murthy. No, there. Arti Murthy is there.
There is one like a query on the Dr. KVK Sirugumani. He is, but I did not see his name here.
No, that's it.
Because he said that I present due to connection problem, but I did not see his name. It is in the Q&A. Q&A, Dr. KVK Sirugamani, he mentioned, but
I don't know. You just check it. Which paper? Title of the paper.
yeah, I did not see any.
Then
I don't
see the title of the paper,
list, sir. So I hope we should not allow.
Yeah, we cannot allow that if he is not there.
Dr. Das, may I suggest that we wait or send a message to Chinnusamy whether he will be joining now or not, because he should go as scheduled.
Right. Yes, correct. He is invited speaker, so he should be there.
Yeah, and I'm sure he would be there. And there is some might problem. If you have his telephone number, just give him a call or send him a message to come and join.
Yeah, that's correct. That's correct.
You may call him just.
Now, excuse me. Is there any message left by him?
No, sir. No, sir.
No. Can you contact Dr. Sen? He was probably maybe some message there.
Sure, sir.
Please.
Dr. Sri Devi, can you hear us? Dr. Sri Devi, you are already online. Can you hear us? Can you unmute your mic and then can you hear us?
right? Sridevi, she is there, but she is not responding. Srideviya Jakkirao. Hello. Nobody is there to respond. What happens?
Sir, I'm just contacting him, sir. Just give me a minute, please.
I think
Because we are running short.
we have Dr. Manohar now.
Manohar, Dr. Manohar,
Yes, sir.
can you deliver your talk right now?
Yes, sir. Yes, sir.
Okay. So, Dr. K.K. Manohar and Yogini Sanbhag, they're from crop science sections, ICRCCARI, Goa, India. Their title of the paper is Response of Land Races and Wild Relatives of Rice from Goa and Karnataka coast for induced soil stress at the settling stage. Dr. Manohar, please.
Can I share my?
Before you start, there is a stipulations. You try to restrict your talk within 10 minutes. Please.
Yes, sir.
Cooperate with us. Okay, please carry on.
Sir, sir, good afternoon, sir. Sir, is my slide is visible, sir?
Please.
Yes, visible.
Respected chairman and co-chair and all the distinguished participants. Myself, Dr. Manohar KK, senior scientist at ICR Central Coastal Agriculture Research Institute, located at Old Goa in Goa State. So my title of the topic is Response of Land Races and Wild Relatives of Rice from Goa and Karnataka coast for induced salt stress at the seedling stage. We all know that in rice, salinity is a major problem after the drought, and the salinity problem is more in seedling as well as the reproductive stage. In this study, we concentrated on the response of the land races and wild relatives of rice, which is being collected from the Karnataka and Goa coast. And we wanted to know the response to the induced salt stress. We have induced the salinity at 12 GSM per meter. Coming to the introduction, as we all know that salt stress is the second most widespread problem affecting the rice crop after the drought. So identification of the novel salinity tolerant sources from the germplasm pool is always very useful for the plant builders to widen the genetic base, because the continuous usage of one or two donors in our breeding program would always lead to genetic erosion. So we wanted to include more the donors so that we can broaden the genetic base of the materials what we had. The traditional the land races and wild relatives of rice present in the coastal and the interior parts of Goa and Karnataka are unique in terms of their phenotype, response to salinity and water stagnations, which is a common problem in these areas, and also the yield attributes. So in this study, we wanted to see how these materials which are collected from the this Karnataka and Goa coast responds to the salinity, the induced salinity under the artificial controlled conditions. Sir, here we have taken up the explorations since 2013 and till 2018 to collect 162 germplasms of rice. In that 126 are land races and 18 are wild relatives, mostly the Vareza nivara types. And we have also included 15 already released varieties at the national level, and also 3 standard checks for conducting our study experiment. So this is the season in the year Kharif as well as Rabi 2020-21. We have conducted our experiment. We included altogether 162 genotypes, and these are the microplots which we have wherein our experiment was conducted, and we have studied the response of salinity stress at the seedling stage. And each tank has got around, we accommodated 43 genotypes in each of the tank with a single row, and maintaining 50 centimeter by 15 centimeter. And FL478 and Pokali we used as a tolerant checks, and IR29 as a sensitive check. And these are the we can see the differential response of the genotypes to the induced salt stress at 12 decibels per meter. We can see the clear-cut differential between the tolerant and the sensitive genotypes. This is one of the genotypes that we have used, that is Govardhan 2, from the selection we made from the popular landrace prevailing in our Goa state, that is Korgut. And also we can see this is the response of the Pokali, an internationally known tolerant check variety for seedling stage tolerance. And this is one of our line KS19-2, again selected from the Korgut landrace, showing tolerance response. So these are the again some of the photographs we had taken the after the uprooting. We have taken the fresh the shoot length and root length, fresh shoot weight and dry root weight and dry root weight, dry shoot weight and dry root weight. All these parameters were measured, and we have made the variability assessment on these characters. This is the photograph showing the sensitive variety, that is sensitive line IR29. All these lines are tolerant lines from our study, including FL478, Pokali, Govardhan 2, Korgut, CSR 27, and our another popular landraces like Red Savani, Karibatta, Madras Sanna, and JK58 and Lusae. And this is the summary of the table, wherein how these tolerant reactions and the moderately tolerant reactions and the sensitive to highly sensitive genotypes showed the values for this is the values for fresh shoot length, root length, fresh root weight, fresh shoot weight, dry root weight, and dry shoot weight. And we also estimated the potassium content and sodium content in the shoot as well. It showed that the tolerant genotypes were showing the very low value of sodium to potassium, indicating that they're excluding more of sodium and intake the more of potassium. The value is 0.81 for Pokali, and it's ranging from 0.7 to 1.13. Whereas in case of moderately tolerant genotypes, also showing 1 to 2 values for sodium to potassium genotypes. However, in the sensitive genotypes, we can see that values are more than 2 to up to 4. The tolerant check variety FL478 is showing 0.7, whereas the highly the sensitive line IR20 is showing 7.242 for sodium to potassium ratio, indicating that the lower values of sodium to potassium ratio in these tolerant genotypes. And we also in the Kharif season we had worked out in the normal field conditions the different genetic variabilities for all these genotypes what we had collected, and they had shown a wide range of variability for all the characters that we have measured, from days to 50% to grain yield. And in we also used the 34 different SSR markers found in the genomic regions in the linkage group of rice to study the genetic variability and the diversity among these genotypes. And it showed, and we calculated the different molecular observations like PIC and other expected heterozygosity. And all these markers have shown high PIC values, and RM10871 showing the highest PIC value of 0.92. And this is the we grouped these genotypes based on the data generated from the 34 SSR markers, and we had seen that 4 different clusters. These genotypes were grouped into 4 different clusters, and these based on their reaction salinity, we can use them in our breeding program as and when it is needed. So finally come to the results. In our study, we had used 162 genotypes, and we got 8 genotypes which are showing highly tolerance reactions to salinity. And as we said earlier, these genotypes would serve as a novel sources for sources as donor in contributing for in governing the tolerance to salinity such as seedling stage. These genotypes can be well utilized in our breeding program for developing the new salt tolerant varieties for the coastal saline areas in the West Coast region as well as in the entire coastal regions of the country. And we also have some moderate tolerant genotype. These needs to be studied further to draw further inferences. With this, I would like to thank sir. I would like to thank the organizers for giving me an opportunity to share my experiences, what we had at Goa. Thank you, sir.
Thank you very much. You kept your time schedule. For that, again, thanks. Now, any questions? Very rapid questions, or anyway, just to, it is a very practical approach to study at seedling stage the salt tolerance identification, and as many as 144 genotypes taken. And potassium and nitrogen, potassium and sodium, that is ratio has been estimated.
Yes.
And then, of course, SSR markers, as many as 34 used, and genetic diversity estimated, and 8 genotypes identified. Actually, I was wondering those 8 genotypes, whether genetically they are very much diverse or they are coming to the same cluster.
Yes, sir. So that would be studied, sir, now again with the help of our results. And we need to identify the loci where they are located, and after that, whether they are of same as that of the FL478 region or some other region is contributing to the tolerance.
Okay.
That will be worked out, sir.
Okay. Okay. Thank you very much. So shall we proceed for next?
Thank you very much, sir.
Yeah, we can proceed for next. But in the meantime, Dr. Manohar, can I raise that? How about, did you check even the reproductive stage? Are they well like seed setting, everything is good, and they are like having a close association between the seedling stage tolerance and reproductive stage tolerance?
Yes, that's what, sir. We have now left it for reproductive stage also. Whatever has been dead, sir, they are they are discontinued. Only these tolerant genotypes are now kept for screening under the reproductive stage, sir. It's ongoing, sir.
Okay, thank you.
Yes, sir.
Thank you. So next, who is next? Shall I call upon?
Sir, can I present
Dr.
Sridevi?
Next.
Sridevi.
Yes.
Yeah, now Dr. Sridevi.
Sridevi Jakkaral. Oh, okay, okay. We are very much trying to find out. Anyway, now you are audible. So, Sridevi Jakkaral, it is a zonal agricultural and horticultural research station. It is Brahmbhar, Karnataka. So, Sridevi, please deliver your presentations, and her title are Submergence Tolerant Rice, Red Rice Variety, Sahadri, Panchamukhi, for lowland situation of coastal Karnataka. So please, Sridevi, please.
Sir, my slides are...
Okay, it is coming.
Yes.
No problem.
Very well.
But please stick to your time. Once again, I remind you.
Okay, sir. Okay, sir.
Okay.
Sir, in coastal Karnataka, we received heavy rainfall in Kharif season, sir. So, and a farmer here in coastal region, they are growing traditional lines, and we all know that they are prone to lodging, low responsive to the fertilizer input, and generally they will yield low. But in coastal region, Karnataka, the MO4 is red rice season, is a predominant rice variety growing in Kharif season. due to this drastic, erratic rainfall, this MO4 is not tolerating this submergence situation. So it is giving less yield under the week-long flooded condition. So this is the reason farmers are getting low return. So this is the situation in our coastal Karnataka, where all traditional variety and earlier released variety, they are non-lodging to the heavy rainfall. And for this background, we identified one red rice variety, that is IRGA 318-11-6-9. It is from Irlana. It is IRRI line, sir, which is selected in 2009. From that, we selected based on the pure line selection to develop the high yielding medium gold red rice variety, which is tolerant to submergence. And it is situation is for the lowland situation. And this is the distinguishing characters of Sahyadri Panchamuki and MO4. So for this line, we named as a Sahyadri Panchamuki. So it has a special 5 characters, that is submergent tolerant. It's situated for lowland region, and this is a red medium bold type. And this is some distinct character. It is a semi-tall, and compared to MO4 is a dwarf, and it is it has some special character. This is the stem thickness is 3 centimeter when compared to MO4, that is a 1.5. Then it is submergent tolerant up to 8 days, and its MO4 is non-tolerant. And Sahyadri Panchamuki is a non-lodging type. And this is our station trial. This Sahyadri Panchamuki is significantly higher yield with 16% increased yield when compared to MO4. And this is our station trial. This is a Sahyadri Panchamuki. And this is a multi-location trial. Here also we'll get more yield with a 21% higher yield. And you can see in the picture, this is a MO4. It is already lost due to flood. And this is our Sahyadri Panchamuki line selected for flooding. And this character we observed, there is a root development at a nodal region. And this is some traditional variety grown by farmers, and they are facing lodging so that there is no yield. And this is our situation where we received heavy rainfall at Kharif season. So it is a Sahyadri Panchamuki is withstanding and giving the good yield. And this is the same situation. And here our Sahyadri Panchamuki is performing better in submergent tolerance. And the same picture here, same. And this is the results of Sahyadri Panchamuki at ASRP trial. The IET number is 27670. And this is the same yield performance at national level at ASRP trials. And this is a disease and pest reaction. It is tested in 32 locations. All the leaf blast, neck blast, seed, blight, rot, BLB, and all the disease were tested along with the stem borer damage, leaf folder, and BPH, WBPH, Galmage. And for all the disease and pest, it is showing the resistant reaction. And this is the physical characteristics of Sahyadri Panchamuki when compared to MO4. So for all the hulling, milling, HRR, for all the characters, seed characters, it is on par with the MO4 only. And here the physico-chemical characters, we only observe the differences is the Sahyadri Panchamuki is take less water for cooking. That is 175 ml per 100 grams, but MO4 take 225 ml per 100 gram for cooking. And there is a gel consistency is Sahyadri Panchamuki is 44 and MO4 is 49. So it is compared to MO4, the Sahyadri Panchamuki is a little bit soft after cooking. And here there is some cooking quality parameters also we tested. So there is an increase in the single seed grain weight. It is measured in milligrams. After cooking only, it is there is a weight is more in Sahyadri Panchamuki, and it has a bulk density. It has a more after cooking. You can see the 1.77 in Sahyadri Panchamuki, and it is a 0.95 after cooking. And we observed some less leach out solids per 10 grams when compared to MO4. And And these are the sensory, means organoleptic evaluation of Sahyadri Panchamuki. So overall quality parameters, this is appearance is good, aroma is little aroma, texture is good, taste wise also good. So overall acceptability is good. So with this find out the bulk density of Sahyadri Panchamuki is heavier when compared to MO4. And this is the zinc and iron content of Sahyadri Panchamuki. So here the Sahyadri Panchamuki has got a iron content is 11 ppm and zinc is 20, and his MO4 is 20 and it is 15. So here we observed zinc content is more when compared to MO4, and these are the some characteristic. And this is the DNA fingerprinting of Sahyadri Panchamuki and compared to MO4. This is the RM258 SSR marker linked to the this is a fertility restorer gene that gives in the same in 2 variety, it is giving the different fragment base length, and this is the IC number we got. And so from 2019 it covered in 500 acres of lowland region where there is heavy rainfall, and in 2019 it covers 10,000 hectares. So these are the some features we have, and this is the seeds of Sahyadri Panchamuki. It is a bright red color seeds, and these are the parboiled rice. We hear in coastal Karnataka, people used to red rice, that is a parboiled rice, and these are the some cooked rice entries, and these are the some photos. So these, thank you, sir.
Thank you very much. A very practical paper, this one. Now, actually, you have referred about the submergence. What is the depth of water? That submergence you are talking about. What is the depth of water? Height of water?
Sir, sir, whenever rainfall we receive in June, July, it's very, sir. The depth is very, sir. When we receive more water, up to 50 centimeters is there, sir.
Okay, up to 50 centimeters.
You can see in one of my slides, sir. Here, slide
Now you have
show
referred. Okay.
Yes, sir.
Now you have referred about that.
You can see in this slide, sir. You can see in this slide.
That's right. That's right. But
whenever we receive rain, sir, we'll up to 50, 10, 50, 60, like that. Up to panicle, up to this panicle length also will receive, sir. That much is there. It is in. It is not constant depth, sir. It will vary.
Okay. Okay. And then you have referred about that less water taken.
Yes, sir.
Could you suggest why and what is the anatomical feature? Is there any particular why it is taking less water?
No, sir. We are not studied that, sir.
Oh. But this is a very simple but useful, I believe, because this is parameters, less water taken under submergence. That could be one of the reasons that it can stand up there. And another is stem thickness.
Yes, sir.
Those two things are very important, it appears. And then, of course, you have done the DNA fingerprinting and and try to find out the differences between this and the mother one. So anyway, it is a very important issue that many times with submergence stress, it happens. And because of uneven distribution of water, and it is very common all over India. So this is very important issue. Dr. Singh, any comment?
Yeah, I would like to ask Dr. Sridevi, that's a very good presentation. And it's as Dr. Das mentioned, it's a very practical, like applied approach. And did you also find that when it is a coastal area, which you mentioned, did it have some salinity also in that water? Was it in the high tides?
Yes, sir. We salinity problem is there, sir. I am doing for that, sir. Some research we are conducting along with ASRP trials.
Yeah, because we need to have like a 2-in-1 varieties which are having salinity plus submergence tolerance. Not alone will not work on that. Thank you very much.
Sir, can I ask a quick question?
Okay, we are running short of time really. Can you, anyway, please do it very quickly.
Okay, sir, I am a
Please.
bit confused, Dr. Sridevi, although the work is good, but then whether it is a submergence tolerance you are talking about or stagnant tolerance or it is a deep water rise. Because one of the slide you have shown the root is coming from the nodal segment. That is the typical symptom of the deep water rise. But 50 centimeter height is not a submergence. It is a stagnant between deep water and the submergence. So it should be, looks to be a stagnant water tolerant rise. That is what I just wanted to.
No, sir, it is not. No, sir, it is not stagnant, sir. Whenever we receive the heavy rainfall in June, July, August,
yes,
there is a some submergence is there, sir. Means there is a flooding situation is there, sir.
Yeah,
in
I
3
know
months.
that, but your picture looks that there is
After
a stem elongation. That is what I am surprising. So stem
yeah, sir, that is
stem elongation is a trait which is not a submergence, not related to submergence tolerance. Stem elongation is related to either deep water or stagnant tolerance, and typically the roots come from the nodes. But in case of submergence tolerance, the plant doesn't grow; it remains under water, and then when the water recedes, then it started grow. So that way the definition is not clear to me. Otherwise, it is a good work, wonderful.
Yes, correct. Actually, Dr. Tapan Mondal is already correct, because in case if it is submergence, there is no oxygen. Even the leaves, every leaf is underwater. And in case of the leaf is out, then parenchymas work for the respiration. So let's see this one, and please take care of that. In case submergence is there, it should be at the earlier stage probably. I don't think that there is submergence on the reproductive stage. Okay. So Dr. Das, I think we have Dr. Dhanush Kodi now, who was the first one. I think we can give the floor to him.
Okay. Please.
Sir, is it audible, sir?
It is audible. Let me. This is a Dr. Dhanush Kodi.
Yes, sir.
Krishi Vigyan Kendra.
Yes, sir.
TNAU, Tamil Nadu. Your paper on evaluation of different salt-tolerant paddy varieties in sodic soil.
Yes, sir.
And farmers' preference for adaptation in Tiruchirappalli district in Tamil Nadu.
Yes,
so
sir.
Please carry on.
So good afternoon, sir. India has an area of 6.7 million hectares of salt-affected soil. In Tamil Nadu, about 4.7 lakh hectares of soil under salinity and sodicity. Out of 4.7 lakh hectares, 3 lakh hectares are coming under inland salinity, and 1.7 lakh hectares are coming under coastal areas. Tiruchirappalli district alone has 11,165 hectares of salt-affected soil. Due to the lack of availability of water, nutrients, and oxygen in the root zones and sodic condition, we found 30 to 50% of yield reduction in rice. At the same time, rice is suitable for rehabilitating saline and sodic soil. But adoption and selection of plant cultivars according to the environment is very important. Nowadays, the modern high-yielding varieties require considerable investment to ameliorate this soil to ensure the reasonable yield under the sodic conditions. Hence, KVK Sirihamani conducted on-farm testing trials under sodic soil with the crop of rice. For that on-farm testing, we selected Manihandam block. That is one of the salt-affected area. The soils of the experimental farms is clear loam in texture with low organic carbon content, low nitrogen, phosphorus, and medium in potassium status. The electrical conductivity of a particular region is 1.9 to 2 decibel per meter, and the exchangeable sodium percentage is 9 to 9.7. Hence, we conducted these on-farm trials in the selected locations with the 4 paddy varieties. That is the paddy BPT Bapatla 5204, that is a farmer's practice. and other three, we compared these PPT varieties with other three salt tolerant varieties like Trichy 3, Gangavadi Sona, and CSR 36. After this trial was completed during 2016, and again the repeated trial was also conducted during 2017. During 2019, we conducted a survey whether the farmers are adopting these salt tolerant varieties through field survey. For that survey, we selected 100 farmers as a sample for sampling. An interview during after selection, the interview schedule was also developed to collect the data on farmers' adoption and influence on farm income. The result of on-farm testing, the table shows that the more yield and highest net return was found in in the field where paddy TRY3 was cultivated, which is followed by paddy CSR 36. The lowest grain yield was recorded with paddy BPT 5204. The difference might be due to the genetic cause and responses of the particular genotype in the soil, and the climatic condition might be reflected in the growth and yield parameters. Similarly, the highest benefit cost ratio and net return was also found with the varieties of paddy TRY3 and compared to other varieties. And on the outlook on the superiority of paddy varieties and farmers' preferences for adoption under the SODIC region was studied. The farmers replied that pest and disease resistance and more grain yield in the salt tolerant varieties, particularly paddy Trichy, and low input requirement and accessibility of seed in the local condition and climatic variation resistance, time of maturity, and the easy of processing during harvesting, and socio-cultural compatibility and market price. Due to these factors, the farmers are preferring paddy TRY3 when compared to other varieties. From that study, we concluded that the location-specific field evaluation of varieties developed at a laboratory level is a need of the hour, and especially in the different salt concentration and field conditions. The result of investigations brought out the higher yield of rice could be achieved with the paddy TRY3 could be due to the salt tolerant capacity of the particular variety and the similar management practices in sodic soil. Similarly, the favorable benefit cost ratio also found in paddy TRY3, which is highly suitable for adoption for enhancing the productivity under soil in particularly Tiruchirappalli district. Thus, it was concluded that the various characteristics of different paddy varieties possessing the salt tolerance, seed availability, accessibility, less pest and disease incidence, more yield, and market price were useful in founding or identifying the suitable variety to the specific location to achieve the our goals.
Okay, thank you very much.
Thank you, sir.
Now you referred about that sodic soil. Could you just elaborate it? What kind of sodic soil there?
Yes, sir. It is sodic soil. The ESP is 39 to 42, sir. This is inland sodic areas. Sir.
Then you have referred about that easy of processing during harvesting.
Yes, sir. Yes, sir. Because grain shattering and other problems also we found, sir. In this variety, it is not there.
But it appears that your variety is very tall.
Yes, sir.
And it is a lodging susceptible.
Yes, sir. Some in some areas, for example, in clay soils, we can found lodging, sir. But other areas, we could not found this type of problem, sir.
Dr. Singh, please.
Yeah, it's okay because I know because I work on the sodic soil, and these varieties are like more or less, having the very high pH tolerance, and because of that high pH, the nutrient availability becomes in the question. Yes, definitely this tall variety at just high pH, it should reduce its height. So I would like to know that when you have this thank you slide, I think this field is not sodic soil. It is a normal soil.
It is a saline soil, sir. For this slide only I used. Previous slides, previous this one, and these areas are coming under sodic region, sir. That's why we applied gypsum and green manure also we grown and incorporated in the trials, sir. The left side you can see the problem of sodic soil, sir.
Yeah, the one slide which you shared, the thank you one, I think it looks like a very normal soil. That's why I was thinking it's.
Yes, sir. That slide is normal soil because this farmer is now now coming voluntarily involving in seed production, particularly in the paddy TRY3. Then he distributing to the farmers, nearby farmers who are affected by the soil salinity and sodicity, sir.
And how about the CSR 36 acceptance?
Yes, sir. The CSR 36 it is also, sir, give more yield, but the local, but we can follow these varieties, sir. But the seed availability in these locations are struggling, sir. That's why they are preferring the local variety which is suitable for the particular location, sir. And
Okay,
another one important, sir, the paddy TRY3 that is suitable for value addition. That is for idli making. You may aware of that idli. It is South Indian varieties. That's why the farmers are preferring this particular variety and compared to other varieties, sir.
Okay, but just to add on this, thank you very much. But add on that, if you have the seed problem CSR 36, you can write to the director of CSSRI Karnal. He can help you. Yeah. Thank you.
Sir. Thank you, sir. Thank you very much.
Okay. Thank you very much. Now, next, who is ready? Next is DP Matra. Is he available?
No, sir. Next is my presentation only.
Yeah, Dr. Savardekar.
Yes, sir.
Dr. Nitish Ranjan Prakash.
Yes, sir. Yes, sir.
Oh, Nitish Ranjan Prakash. Okay. Let us see. Please.
Is he available, Dr. Nitish Ranjan Prakash?
Myself itself, sir. I am only Nitish.
Oh, okay, Nitish. Yeah, you told me Prakash.
Okay,
Prakash and Nitish Ranjan Prakash, it's same. Yeah, okay, thank you.
Okay. Okay. Let me tell you that he is man from ICRC SSRI, Karnal, Haryana, India. So he is from Karnal. Please, A.N.R. Prakash, Nitish Ranjan Prakash, please.
Good afternoon, everyone. I am Nitish Ranjan Prakash, working as scientist here at ICR Central. Sir, my slide is visible?
Yes.
Very well, but we'll request other people to mute the mic who are not speaking. Thank you.
Yes, sir. So, myself, Nitish Ranjan Prakash, working as scientist, genetics and plant breeding at ICR Central Soil Salinity Research Institute, Regional Research Station, Canning Town, West Bengal. I'm going to present today on my topic, evaluation of rice genotypes for tolerance to seedling stage salinity tolerance under hydroponics. This research was part of my PAT research project as a part of ARS training. I have recently joined here. So,
Dr. Prakash, can you make full screen, please?
Sir, actually, it's in PDF mode, and the system that they have given us to just.
Okay, please go ahead then.
So, in that, PowerPoint is not there, so that's why I have to struggle. If I have to go, means I have to close this session, then only I can do something.
Okay, no worries.
Okay, sir. Sorry, sir. So, as before going on to presentation, I want to bring your kind attention onto rice, which is a master crop of all coastal regions, and it is India is second largest producer of rice. It is largest exporter also. But the problem we are having in coastal region, like we are having of waterlogging tolerance and low productivity, because of we are growing mainly the local varieties that we are growing mainly in coastal regions, as I can see in Sundarbans. And coastal regions are also being affected by the rising sea level, increase in temperature, and these all things. In general, the genetic study for salinity tolerance in rice is having these such traits like starting from dry mass, biomass, and root and shoot sodium potassium concentration, and root and shoot length and seedling length, and these all things. But while at reproductive stage, we are mainly focusing on the mainly focusing on the number of grains per panicle, number of fertile specklet, pollen sterility or fertility, 1000 grain weight, etc. And so in the present study, we went on with 32 rice genotype. We had grown them in glass house, and salt treatment was given on 14th day as per the ERI manual, and the sanitary stress was given at the rate of 10 decibel per meter, and data were recorded on SES scoring, root length, shoot length, and SPAD chlorophyll reading. And based on root length, best genotype was Agami and poor genotype was I29. Based on shoot length, the best was Nonobokra and poor was I29. Based on plant length, the best genotype was Nonobokra and poor was I29. Based on shoot length and root length ratio, we could find that Biridhan 78 was the best genotype and VSR 156 as the poor genotype. Based on chlorophyll content, FL 478 and was the best, and I29 was the poorest. So we were able to find that all the genotypes were having significant difference for all the traits that we have studied, and we could find that salt evaluation score or SES score was negatively correlated with chlorophyll content, root length, and plant length, while not with shoot length. Based on all the morphological data, we were able to cluster all the salt tolerance genotype into a kind of single cluster, and this study was a part of like metacutal study that I had performed there, and we could able to also able to predict some metacutials, and that we are currently validating now. And based on this study, I29 was the poorest genotype, as we are also using it as a check in our most of the experiment for screening, and FL 478 was the best, and the presence of genotypic variation for tolerance to seedling and reproductive stage salinity is common. And the negative correlation of SES score with shoot length, root length, and chlorophyll content again describe its accuracy in identifying the salt tolerant genotypes at seedling stage. Morphological traits such as root length, shoot length, chlorophyll content, SES score are able to classify genotypes into different clusters, and interestingly, we can cluster known salt tolerant genotypes in one cluster, but not others. It again speculates the hidden unexplored source of variability in genomes of different cultivars, which are contributing towards salinity tolerance. Thank you. Okay, sir.
Thank you, Dr. Prakash. Yeah. Dr. Das, your mic is mute, please.
All right. 32 genotypes you have, yes, right?
Sir. Yes, sir.
And your standard is a Nonobokra, right?
No, FL 478 and Pokali we had taken, sir, as a standard.
Pokali also you have taken?
Yes, sir. Yes, sir.
Okay, okay. And then you have made some physiological and some morphological character study. Now, in that case, you have given much more stress on chlorophyll content, right?
Actually, sir, we had tried to evaluate these genotypes for mainly for like metacutal part, but because of time constraint and these all things, I could not present those things in a kind of that one, because it was in.
Okay. Have you taken the stomata?
No, sir.
No.
So we have just taken only chlorophyll content based on a SPAD reading only, not for estimation from that reason.
Anyway, Dr. Singh, please.
No, it's okay. I think it's a preliminary study. I think he has to go ahead with the little bit more different, more study. Then only he can be having the concluding something like, you know, in a better way.
That's right. Next, who is there? Dhanush Koti, is he there?
Next is SV Savardekar, sir.
Yes, sir. Yes, sir. I am here. Yeah, I am here, sir. Yes, sir.
Just a minute. Just a minute.
Yes.
Who is there?
Dr. Savardekar.
Swardekar. Okay. Please, Dr. Swardekar. Just a minute.
15 number.
Number 15.
Savardekar Sahab, namaskar.
Sir ji, namaste, namaste. Nice to see you after so many years.
Oh, okay. Oh, that's why because first day was the Mohitra, but that's okay. So you are from Plant Biotechnology Center, College of Agriculture, Konkan, Krishi Vidyapit, Dr. Balasaheb Swant, Kankar Krishi Vidyapit, right? And it is from Dapoli,
yes,
Maharashtra.
Yes, sir. Yes, sir.
Now you are doing the DNA barcoding of rice varieties developed by your university. So it is very interesting that DNA barcoding. That's very good. Please carry on.
Sir, my screen is visible?
Yes, yes, please.
Sir, first of all, respected chairman, sir, then the Dr. R.K. Singh, sir, and the other fellow scientists. So first, I am very much thankful to the society for awarding me the fellow of the society. So last 35 years, I am associated with the society, and the society is doing very well today also. Before starting my this presentation, so I am very much thankful because my first award I have received by the auspicious hands of the Dr. J.S.P. Yadav, that is the Best Research Paper Award. So I am very much thankful to the society for giving me this second consecutive award. And after that, I am starting my presentation here. So the title of the presentation, it is the DNA barcoding for the identification of the rice varieties developed by Dr. Balasaheb Swant, Konkan Krishi Vidyapit, Dapoli. Sir, as we know, the DNA barcoding, it is a technique for the characterizing the species based on the short DNA sequences of the particular genomic region. Sir, this Balasaheb Swant, Konkan Krishi Vidyapit has developed near about the 35 varieties. Among the 30 are the state varieties and the 5 are the hybrids. Within that, we have developed the 3 varieties, which is the salt tolerant, that is the Panvel 1, Panvel 2, and the Panvel 3. So here we used the we have developed the passport data of all this variety. We also have the morphological characteristics of this variety, but the DNA barcoding, it was lacking, and that we are trying to do here. So the genesis of the study was to develop the molecular tag, because there are the number of varieties we have developed. Some of the varieties, they are the phenotypically very same, but they are the genotypically very different. So what are the variations are there in that particularly varieties? That varieties that we are going to identify here. And earlier we have done the number of the studies has been based on these nuclear markers, but now we are concentrating on the chloroplast markers. So here they particularly used for the reviewing the phylogenetic relationship among the related crops. So here, if you go for this, so there are the chloroplast maturus K, that is MATK, is one of the most variable coding regions of the angiosperms. And here we are going to use here the 7 candidate plastid regions. That is, we have given the list of the markers also. And based on this, we are going to identify which is we are going to tag this particular variety, because there are the problem of the biopiracy also. So this particularly we are going to solve with the help of this particularly barcoding. As usual, we have followed the methodology. We have gone through this, and here the J dilution and the PCR product we have done. We have done the sequencing with this particularly outsourcing, and at the same time the data analysis were used with the bioinformatics tool here. So this particular methodology I have given here in short. So these are the 10 barcoding primer, that is the primer pairs we used here. And almost all these are some of the plastid genomic sources, and one it is only the this is the nuclear source. So this particularly this MATK, then the RBCR, we know that particularly this region of the chloroplast gene, particularly RBCR, that is Rubisco large subunit, is used for the barcoding of the number of the plants. So we are also tried here by using this. So that the present investigation we made the first time to characterize the varieties developed by the DBS-KK with Apoli. At the same time, we used the SSR primers also, so that we can able to characterize that particular variety which is carrying that particular specific allele, particularly for the drought or the salinity or the BPH or the blight. So these are some of the bioinformatic tools we have used here, that is multiple sequence here using the MEGA 7.0.25 input. And then we also the molecular evolutionary genetic virus, that is the MEGA 7. Then we are used with the sequence polymorphism analysis. And then we use for the visualization based on this particularly the software, that is the fingerprint. So these are some of the experimental results of this. So we are divided into the three, that is the GLA analysis, then the MEGA environments, and the barcode generated. So we have taken here about the 20 varieties here, but among these the only 16 varieties are shown the amplification. And here the range of the amplification and the concentration of the DNA, it is also given here. We have measured that, and at the same time the range of the concentration of the DNA, it is also given here. So because we face the problem of the elution, whenever the concentration of the DNA it was less, it was not getting eluted, and at the same time the number of varieties which is there, we cannot able to sequence it. No doubt it was our first study, so that we have whatever the material was there. So in that material we are trying to develop the barcodes. So here the MATK one. So this is the one particularly the primer sequence, and these are the particularly the chloroplast maturus K, which is having near about the most variable coding genes of this rice. So here we have generated the barcodes into this particularly the Karjat2 and 2 and Karjat1 and the number of. So that means the three barcodes we have generated here. And here you see the mega alignment. So this is one, it is a standard, and based on that we have done the mega alignment of this, and we have developed. And these are some of the barcodes generated by the Karjat2. This is for the Ratnagiri 2, and these all the barcodes which are generated for the Karjat1, Karjat2, Karjat4, Ratnagiri 5, Ratnagiri 1, Ratnagiri 3, Ratnagiri 4, and the Panvel 1. So like that. So this is some of the I have made here a very clear picture that. So these are some of the standards are there, and with the help of the standard we have compared this, and this particularly they are having this particular sequence, it is absent in case of the Karjat 1, and based on that the groupism has done here. If you see here in the Ratnagiri 2, it is coming into the another group. So like that, based on this color and we develop the barcodes. Now we can able to identify our varieties. So this is another one, that is the RBCR. This is particularly shown the amplification in the 831 to the 884 base pairs here. And here we have generated the barcode of this particularly all samples except Karjat 1, the barcode it was the same. So here we have developed that. So like that, this is the TRNK. This is the particularly the barcode which we have developed. So these again 4 we have generated the barcodes with the help of this, that is Karjat 4, Karjat 7, then the Karjat 2, Karjat 3. This is the one in one group, and based on this, so we have developed the phylogenetic tree also. We have based on that we have done the groupism also. Then the NDHA. So this is the particularly the primer pair we have used here, and here we have developed the near about the among the 20 varieties, the 19 varieties shows the amplification. And this particular the DNA quantity was among the 16, it was absent. So these are the way of generated the barcode of this. Particularly the variety developed on the barcode. So like that, there is a combination of the MATK1, RKIM, and the MATK3, FKIM. So this is the one. So here there are the three groups we have generated, that is Karjat 7, Panvel 2, and the Palghar 2. So like that, these are the two variable groups are there. At the same time, you see here, based on this standard, we have developed these barcodes. So among these 10 barcodes, we have seen the efficiency of this particular barcode. So there are these varieties shown in the application. There are the 100% barcode. It has been generated by the PSBA dash TRNEH. So they are shown the 100%, then the TRNEH and the PSBA, and there are another one that is the TRNK. So these particularly markers we identified, they are generated the 100% barcodes. So they are sequence number, the number of sequences varieties obtained here, the different primers, they have shown the different sequences also. So these are the total 16 varieties we have taken here. So these are some of the experimental results with the help of the molecular markers, that is SSR markers, which is commonly used here. But this particularly were used for the identification of the specific alleles, which is carrying that. So here among these, so these are the 7 markers we have identified, and these are shown this 100% polymorphism by the 3 markers. So like that, we have developed this polymorphism, and here with the help of this, we identified the dot. This particularly, these are the varieties. They are carrying the link. They carrying the allele of this particularly for the dot tolerance. Then the Saltal also, Panvel 1 and the Panvel 2 we have taken here. Then the PPH also, they are carrying the allele of the Kurzet 1, Kurzet 2, Kurzet 3 and the Panvel 1. And the BLB, these are the 4 varieties they are carrying the allele. So this particularly study will be helpful generally for the gene editing also. And the same time, whenever you are going for this particular the crossing program, so these are the, and most important, that is the biopiracy. So we can able to protect our varieties now. So thank you. Thank you for your patience listening.
Thank you very much. Now it is very interesting that as in a concluding remark you have said about the bioprivacy or piracy. That means privacy safeguarding. Now it is important in the sense that we have to fight in international arena for our genotypes, right? Now,
yes, sir.
I was one, that's right. Now, actually you have done in your material and MATK, that MATK, amplification, but comparing MATK, other 2 you have suggested, they are better, they are 100% you are showing. Okay. Now, do you suggest then,
yes, sir,
instead of MATK, we'll go for those 2?
Sir, it's not like that actually. So there are the number of markers because and it depends upon the means varieties also. Because that particular sequence, it is not ascent, that means it will not show the amplification.
Okay.
So it doesn't mean that this particular all these markers are useless.
Then you have said about the SSR markers. How many SSR markers?
Sir, we have actually, sir, we have used near about the 50 SSR markers, but I have just only given here the 10. So which has shown the 100% the polymorphism. Yeah.
And what is the number of alleles? Alleles you have allelic polymorphism.
So they are actually each particularly allele, they are identified 1 or 2 alleles only, not more than this.
Okay. Okay. Now, can you locate how far they are, or they are clustered together, or they are little bit away from each other? Alleles?
No, sir. No, sir. We are going to do the further study in the next phase.
Okay. Okay. So that's okay, but it is basically plastic DNA, right?
Yes, sir. Yes, sir.
Plastic DNA.
Yes, sir.
So what is the difficulty for your nuclear DNA?
Actually, the nuclear DNA, sometimes there may be some of the changes are occur. But in case of this particular plastid or the chloroplast or the mitochondrial DNA, they are stable DNA. Stable.
Okay. Okay. That's
genome
right.
is stable.
Right.
Yeah.
Right. Okay.
Yeah.
Thank you very much, Dr. Singh.
This is very excellent study. I would like to know, because I am working in the different environments now on the crops which are not having any reference genome and not many markers also established like SSR. So can you do it on those crops also where there is not much markers available, like or maybe no reference genome? Can you do the same study with that also?
Yes, sir. With the help of the comparative analysis, we can able to do this.
Okay. Okay. Thank you. I will contact you in that way.
Yes, sir. Yes, sir. Definitely, sir. It's nice to you, sir. Thank you.
Thank you,
You were talking about bioinformatics.
Yes, sir. Yes, sir.
So
Yes,
probably bioinformatics will give you some more ideas about this, how many SSR and all these things, right? So
yes, sir.
that's
Yes, sir.
it. Yeah. Okay. Thank
Thank
you
you,
very
sir.
much. Thank you very much.
Thank you, sir. Yeah.
Okay. Then again, Dr. V. Dhanushkodi, is he there?
Dr. Veena.
Veena,
I'm Dr.
Acha, V. Okay. Is she? Okay. Actually, written as a V, so we couldn't
Sir,
figure it
sir,
out.
Sir,
Okay,
am I
that's
audible,
fine.
sir?
Yes, you are from Krishi Vigyan Kendra. Oh, not which?
No, sir. I am from Kerala, sir. Rice Research Station, Vaitla.
Oh, just a minute. Just
Number 16.
Number 16.
So, is it possible to share my slides from your side, sir?
No, you can show your side from your side. Veena, okay. Oh, this is SSR marker analysis for the identification of the elite rice variety, Lavanya, with the parent BTL3 and DNA fingerprinting. So this is another SSR marker analysis. Okay, please, you carry on, please.
So, due to some technical problem, I couldn't share my slides, sir. Is it possible to share it from webinar site, sir?
Only they can tell, organizer.
Nitish, can you do it?
No, sir. Actually, sir, from our side, no, the PPT is not working in this computer by mistake.
So can we do one thing that in the meantime you can set like a fix the problem, whether Dr. Veena or you, and in the meantime we can go to the next speaker.
Okay, sir. I will try, sir.
Yeah, please.
So next speaker is Arte Maruthi. Arte Maruthi is there. Dr. Maruthi.
I'm going to share this.
I'm audible, sir.
So can I proceed with Dr. R. M. Maruthi?
Yes, sir.
Okay. He is ICR Kraisab, Barakpur, Kolkata, India. It is his paper on a GIS-based diversity assessment of Indian Sanem accessions for enhanced biomass and fiber yield. So, Dr. Maruthi, please.
Sir, good evening to all. Am I audible, sir?
Yes, please.
Okay, thank you. Sir, myself, Dr. Maruthi, sir, scientist, crop improvement division from ICR Kraija. So I will be presenting my topic on GIS-based diversity analysis, diversity assessment of Indian Sanem accessions for enhanced biomass and fiber yield. So basically, I will just in a brief slides, I will give about an introduction about Sanem as a fiber crop. Sanem is an annually renewable lignocellulosic non-wood biased fiber crop, and generally for fiber purpose, it will be sown in the month of April. In about 90 to 95 days, it will be harvested and rated for about 24 to 48 hours, and it gives it occupies an area of 0.3 lakh hectares with a production of 18,000, approximately 19,000 tons. The productivity is around 6 quintal per hectare. So in India, means generally for fiber fiber purpose. 1 second. Hello. Okay. Generally for fiber purpose, it is cultivated in Bihar, West Bengal, Madhya Pradesh, Maharashtra, Rajasthan, Orissa, UP, and Tamil Nadu. So it covers nearly 90% of the total cultivated area. And India is the third largest producer of Sanem fiber, followed by Bangladesh and Brazil. So it's just a pictorial depiction of the Sanem. Sanem. It is a fully grown crop at 90 days. So after that, it will be harvested to base, and it will be bundled into suitable size. And after about one day, it will be dipped in a water for about 24 to 48 hours. And after that, it will be taken out, dried, and fiber will be extracted. This is a procedure. So coming to applications, the fiber has a diversified applications. It is used for the preparation of rope, tissue papers, different quality papers, then fishing net, as a canvas preparation, and as a floor mat preparation. So apart from this, as a novel or a new application is it is found suitable as a biofuel feedstock. So in future, in next slides, I will be elaborating this part. So apart from the conventional application, it has evolved an annually renewable non-food stable substrate for biofuel production. Since the fiber contains a high percentage of cellulose, it is approximately around 75% cellulose and 10% hemicellulose and 10% lignin, with high crystallinity and degree of polymerism. So polymerization. These are the two traits, that is high crystallinity and degree of polymerization. These two are the very crucial traits for catalytic hydrolysis of fiber into bioethanol. So in a recent study in a IIT Kharagpur, they have found that through microwave assisted ion liquid mediated rapid crystalline conversion, in just about 46 minutes, the fiber can be converted into biofuel. So this is the new area is developing. So using these new techniques, the biofuel production from non-food lignocellulosic feedstocks like SunAmp has a potential to replace significant percentage of fossil fuel, mainly because SunAmp is being a rapid growing, high biomass yielding crop, and it takes very limited time. Within 90 days, it reaches it produces nearly 15 tons of green biomass, and it requires minimum input. And this solves the problem of food versus fuel debate on over biofuel production. So keeping these things, and further, in spite of having enormous industrial applications, the area under this crop is drastically reducing. And one of the reason being the non-availability of consistent high yielding varieties. So in order to overcome this, we need to redesign the breeding program using newer and diverse germplasm to improve the yield and quality of SunAmp fiber. So as a preliminary study, I have taken this present study to understand the existing variability of Indian SunAmp accessions for enhanced biomass and fiber yield, and to identify the geographical areas with high diversity using Diwa's approach, so that we can target those particular areas for a future exploration program and conservation programs. So here we have included nearly 42 indigenous SunAmp accessions collected over 10 different states of India. So using the passport data, that is latitude data, the genotypes are plotted in a map. So in a preliminary statistical analysis, we have recorded plant height, BD, basal diameter, green weight, green biomass yield, then fiber yield, then stick weight. These are the 5 characters we have recorded on these SunAmp accessions. These in a preliminary statistical analysis, we found significant diversity in for all the traits under investigation. So when the performance of the accessions were checked about the compared with the check, that is K12 black, so we could identify some promising genotypes. The genotype with high plant height and bulk density is generally preferred, because since SunAmp means fiber is a flowing fiber, the increase in plant height and increase in diameter ultimately or indirectly yields to the fiber yield. So then again, the accession with high grain biomass and average stick yield are generally preferred, because these indirectly leads to the high fiber recovery percentage and high fiber yield. So these are some promising lines that are selected. To our surprise, whatever the lines like SAN 7, SAN 6, SAN 28, and some other line like SAN 11 and 15, these lines recorded superior performance for majority of these traits, and all these germplasms are collected from Rajasthan, from across districts of Rajasthan. So through next further, we have done a cluster analysis using Manhattan distance matrix and average linkage method. So these 42 accessions were divided into 3 broad clusters. So among these clusters, the accession SIN 10 and 12 from Rajasthan, SIN 27, 35 from Jharkhand, and SIN 24 from Kerala formed a separate third cluster and distinct cluster. Then the accession SIN 6, SIN 7, and SIN 11, another accessions from Rajasthan, and SIN 2 from Kerala. These form the third cluster, second cluster. So in many other accessions did not follow any symmetrical pattern in means with respect to origin and other things. So they grouped in a broader group. So this cluster 2 and cluster 3 are recorded to be divergent clusters, and the accessions can be selected from these clusters for intermating and for development of population. So this is a JS map, grid map developed through by plotting the passport data along with morphological data. So in the first diagram, this is the first diagram where we have calculated the CV for each trait, and we average the CV of five characters, and it is plotted in a JS map. So through this JS map, we can say the accessions collected from eastern Rajasthan, western Jharkhand, in the border areas between Bihar and Jharkhand, they are rich in diversity. They are showing high morphological diversity. Further, a grid map for separate a specific trait like green biomass and fibril were also generated, and the locations with highest value for these two traits were also identified, and future thrust will be given to those areas for future exploration activity, future exploration programs. So to summarize, we found significant phenotypic variation among our study material, and particularly accession SIN 6, 7, 11, 15, and SIN 22 were found to be promising to be included in our breeding program. Then the cluster analysis grouped the 42 accessions into 3 broad clusters. Then through JS mapping, we could identify the eastern Rajasthan, western Jharkhand, and in border area between Bihar and Jharkhand are highly divergent, or the sites of higher high diversity. So that's it. Thank you, sir.
Thank you very much. This is a fiber crop, Sanheim, but quality-wise, Sanheim is inferior to jute, right?
Yes, sir. Yes, sir. Yeah.
That's it.
Well,
and then, of course, you have suggested about the use of biofuel. That is a very industrial application. That is probably many of the this type of crops, people are thinking in that way. Then you have taken 42 accessions, right?
Yes, sir. Yes, sir.
And you find that different geographical distribution, no relation to diversity. One, and then you have suggested location or site-specific diversification.
Yes, sir. Yes, sir.
Now, considering this site-specific diversification, which particular area do you suggest should be investigated further, explorations and other things?
Sir, in our limited available germplasm, we found this Rajasthan part, means genotype from Rajasthan part, they are contributing, means they are taller plant types, sir, with limited branches. So more scope should be given to that particular area to collect more germplasm, sir.
But you also suggested about Jharkhand. Jharkhand is very close to your place.
Yes,
so that is also another scope,
another
right?
area. Yes, sir. Yes, sir.
That's right. Now, about these biofuel applications, I mean, you are suggesting the whole plant or the stem or leaf? What should be the?
Sir, actually, sir, once again. Here, sir, the fiber content, sir, generally means fiber is rich in cellulose, though since it's a polymer of glucose, means fiber is more preferred, sir. not the leaf, sir.
Okay, so that means whole stem
Yes, sir. Whole stem, whole stem.
devoid of leaf.
Leaves, yes, sir.
Whole stem you can try for biofuel applications, right?
Right, sir. Yeah.
But if you want to improve upon the fiber quality, what should be your point? What should be
Sir,
your directions?
fiber quality and biofuel are two separate areas, sir. For fiber quality improvement is towards means it's an industrial application in other diversified uses like sacking, clothing.
Yes, that's
and
right. But
yes,
what should be your aim? Because looking at this quality parameters here, 75.6% cellulose and on hemicellulose, all these, and high crystalline. And what are the parameters you will hit upon, which will give you better quality?
Sir, actually, main point is Sun-Hemp is having 10% lignin. So we need to target the lignin percentage, sir. We need to reduce the lignin content. So we need to identify the genotypes which are having low lignin content. So that's one of the areas, sir.
And again, there is a problem of that, creeping habit. If you are too
less lignin, yes,
drastic,
it won't support the
action.
yes, sir.
Yeah, that's right. Okay. Anyway, thank you very much. Dr. Singh?
No, it's okay, sir, from my side. Thank you.
Okay.
Sir, actually,
yes, please.
Now I hope we have to close the session for now, because we have like another concurrent session in this session only. So for Dr. Veena, we will be taking her presentation at evening session that we are having at around after 5 o'clock. We will take in that.
So I will complete now, sir.
No, madam. Actually, it will be difficult for us now.
Okay.
Veena Devi, it is not possible for you?
No, sir. No, sir. Actually, for us, it will be difficult because we have another concurrent session now.
Okay. Right. And what about the Chunasamy? He is going to present?
No,
when?
sir. No, sir.
He is not going to present?
No, sir. No, we will confirm from him. Try to contact him, but we were not able to contact. We will try and confirm from him. Maybe in the evening, if we can, we will do.
Okay. So we'll close right now?
Yes, sir.
Okay. So it is my humble duty just to say everybody thank you very much. And on my behalf, obviously, I'll be very happy to say that organizers, they have done splendid job. And I'm very happy that Dr. Singh is always there with smile. And that is very supportive gesture. And I'm very happy to be with you, to associate and to share the views. And all the participants, the speakers, they tried their best to be in time, and they have done excellent job. Thank you once again. Thank you. We'll meet at 5?
Yes, it's around 5:40.
Oh, we'll meet at 5:40. Just tell me. 5:40. Okay.
Okay, sir.
Okay. 5:40. Okay.
Thank you very much. And then thanks to Dr. Das, Professor Das, actually, for the chairing the sessions very efficiently and also taking out the summary of the presentations. Thank you very much, sir, for this one. And I think I'm also thankful to the organizers, who made this possible. Something which is out of the world 1 year before, but now it's a virtual reality.
Thank you very much.
Thank you.
Okay, sir. Thank you, sir.
Thank you.
Okay.
Thank you very much.
So again, we'll meet at 17:40 hours, right?
Yes, sir.
Yes, sir.
Thank